Hello, good morning everyone. That's great. Fantastic. So, welcome everyone for the second day of the Tribute Symposium for Euan. And we had a fantastic day yesterday. And looking at the speakers list, it's going to be another stimulating day today. So I'm Bhuvaneish Selvaraj. I'm from University of Edinburgh, and I've been part of the EMC family since 2014. And before we start the session, I just wanted to say a couple of things about Euan and his family, what they've done, particularly the impact they had on the early career researchers, including me, when I started my career here in Edinburgh. Many of them have said about how Euan's humor and wit, but he also had a very sharp scientific mind. I remember in 2014 when I started doing CRISPR and genome editing, and we had some wonderful deep conversations about the genome editing and how the impact of it on gene therapy and things like that. But also the second thing what I observed really well from over the last decade or something, because I had the privilege of meeting them on several occasions, is their presence at every EMC gathering, every enrolling open day clinic, whether it's a rainy day in Edinburgh, they were there. And what it told us is their vision and their commitment to see this EMC center to succeed and everything. So it's a great privilege to be part of this team and working towards a common goal. So let's start with today's academic session. So just a disclaimer, if this session doesn't go well, it's a fault of two directors, Giles and Siddharthan, who dragged us out for a late night drink last night. But I think we've got a terrific session. So there's going to be two plenary talks and then a series of emerging leaders from the EMC. So the first speaker today is Professor Chris Shaw. Most of you know it's a real pleasure to have you here and host you here for giving you a talk around gene therapy. And most of you know Chris has some seminal discovery, particularly around identifying the genetic mutations in TDP43 and FUS, which has opened up a Pandora box and given us an opportunity to tackle this disease. And he's also pioneered in stem cell disease modeling and developing various animal models and stuff. Lastly, his brainchild is this spin-out company, Aviada Bio, where he's taken it further around gene therapy, and he's going to talk more about the progress in gene therapy and where we are. Thank you, Chris.
Thank you, Bhuvaneish. Hopefully everybody can hear me. Yes, I blame the second whiskey. So these are my disclaimers. Hopefully my slide will work. No, it's not working. Yeah. But I would like to mention Ian Wilmot, who was my first collaborator in Edinburgh, dragged me up to Edinburgh. In fact, I met Donald and Euan many years ago before Siddharthan even, and they were talking about the opportunity to build something in motor neuron disease. And obviously, Richard gave a very good account of what was really happening on the ground, and I was a bit surprised to be invited. But Ian really is a remarkable scientist, and he's a very, very modest man. And when he invited me up to talk and to discuss science, he invited me to stay with him. And I discovered then that when his mother-in-law developed Alzheimer's, he bought the house next door and knocked a hole in the wall so that he and his wife could care for her. And that's the kind of man he was. Anyway, so he asked me about who in the UK was a smart clinician, scientist with an interest in stem cells, and he was the director of the Regenerative Medicine Centre. And I said Siddharthan Chandran. And so I have to apologize to Edinburgh for that. And then, of course, Siddharthan has spread its wings and recruited some really fantastic colleagues to build neuroscience here, particularly around ALS, MND. And so I should apologize to Scotland for that contribution. And now he's become, of course, the director of the UK Dementia Research Institute, so I probably have to apologize to the whole of the UK neurodegeneration field for inflicting this on him. But anyway, he has been a fantastic collaborator, and indeed Andrea, who's speaking after me this morning was the first PhD student that we kind of worked together on, and what I discovered from my time working with Siddharthan is that he's able to do experiments far better than I ever could. So most of the good work we've done in the stem cell front has been in collaboration with him. So today I'm going to talk about gene therapy, and I don't need to talk about ALS. We've heard a lot about that, but probably less has been discussed about frontotemporal dementia. These two conditions are connected, although they sound very different. And indeed, if you're a clinician, you might be in a dementia clinic and you wouldn't necessarily think about a motor neuron disorder, but indeed, many of our patients with motor neuron disease have cognitive issues. And I think Sharon Abrams, one of my very dear friends from Institute of Psychiatry days, was one of the first people to really map out the kind of deficits that can occur in that disease. And what we know now about the pathology of these two conditions is that they are connected. TDP-43, which you've heard mentioned quite a bit, it is the dominant pathology in ALS, but also about 50 to 55% of patients with frontotemporal dementia have TDP-43 retinopathy, so the tau negative ones. And many of the genes turn out to be connected. So the story of my interest in ALS dates back to arriving in Cambridge. The way I got there was quite extraordinary. I'd done some work on multiple sclerosis in New Zealand in nights and weekends and managed to publish a few minor papers. And then probably by some clerical error, I managed to get a Wellcome Trust fellowship and ended up in Cambridge with the same supervisor that Siddharthan had. And he wasn't really that interested in ALS, but I was out in clinics in Bury St Edmunds, and I met two women who had developed symptoms of motor neuron disease. And what they told me was that they knew what was coming because their mother had had it. And it was the first time I realized that this could be a familial disorder passed down generations. They had it, and they knew that their children were now exposed. And their really terrible dilemma affected me. And really from that time, I've worked towards trying to understand why motor neuron disease occurs and whether we can do something about it. So that's my story. And this was the first gene that was discovered. This is the first patient we had from Bury St Edmunds. And I worked with a lovely man in King's College London. And at the end of my time in Cambridge, he said, "Would you like to come to King's?" And I arrived there in '95, so that's 30 years. I've been in the same place at King's College London. Anyway, so we struggled to identify any genes, and there was a very long time in which I used to give talks about the genetics of ALS and say sod all so far, because SOD1 was the first gene to be identified, but we were very fortunate in identifying another important variant. And this man, Jimin Sridharan, helped me with this work. He was a PhD student at the time. It did cost me 3 points on my license, I will say. Anyway, we were out collecting DNA samples from this particular family, which had a history of motor neuron disease, and we were able to show that a particular mutation in TDP-43 co-segregated with disease in the family, and we identified other mutations as well. And then shortly after that, we identified mutations in the gene FUS. We'd identified the locus some years before, but these two genes, the two proteins, have a similar role in terms of regulating RNA transcription, splicing, trafficking, and translation. So that was an important advance. And then, of course, the whole genetic world exploded in terms of rate of discovery, and subsequently we've been involved in a lot of gene discovery. So these are different genes. In the blue and red balls are genes that relate to ALS, and the green balls are FTD genes. Some of them are the same, like C9orf in particular, but actually most of the genes discovered in ALS also appear in patients with frontotemporal dementia, so there's an awful lot of overlap. And the ones with green dots are the ones that have actually come from our lab, at least in part, and then the ones with red stars are those that we've collaborated on. And we've managed to identify the causative gene in quite a large number of people, not just people who have a family history, and we're up to over about 70% of those families we can give an answer to, but the same genes are present in people who have no family history at all. And just recently, we've managed to get approval from the Genomics England to sequence genes in people who've got no family history, because there are now therapies that I'm going to talk about later that actually could be applied for people who don't have a family history. Similar sorts of discoveries have been made for frontotemporal dementia. Anyway, the idea here is that you can map out the pathophysiology. You've got all these jigsaw pieces of molecular signals from the genetics to look at disease processes and mechanisms. They start to come together around certain pathways, and these are informative. And so I tried to sort of map this out. This neuron looks a bit more like a squash frog, but it's really talking about the key pathways that are relevant to that dysfunction leads to TDP-43 misaccumulation and disease. And this could be difficulties in terms of nuclear import from TDP-43, its turnover by protein recycling machinery, ubiquitin proteasome and autophagy, and axonal transport. Those are some of the key areas that are involved, and also energetics. So what do we do with that information? We make cell lines, we make IPS lines, we look at people's brains, we make flies, fish, and mice, and we've done all those things. And the idea here is we make some assays that'll tell us whether we've got compounds that might work to reverse these processes, and we do this in-house, or we might do it externally, and of course then we test them in animals, and then we bring them back to clinical trials, and that sounds wonderful. And I spent most of my life identifying these genes, generating cell lines and cell models and mouse models, and distributing them freely throughout the world. Never asked for an MTA or any particular acknowledgement. But unfortunately, not much happened. We didn't get any new medicines for ALS and FTD, really, that were shown to be efficacious. And so I feel there's a big problem that has been lost in translation. We've not really taken these discoveries through to our patients. So this was troubling me about 10 years ago. Published quite a large number of papers, and after you've done a few hundred, it's less rewarding to get another paper published. So we started thinking about developing therapies ourselves, and I met this man, who is Doddy Weir, and this was in one of Richard Scott's diminished houses, council slash, and he had recently been diagnosed with motor neuron disease. And he asked me, how long have you been working on motor neuron disease? And I said, 25 years. I was very proud of this fact. And he said, what have you been doing, man? And that's the moment that he said that, I think. Captured by the Telegraph photographer. And it was really saying, come on, you've made all these, you're very pleased with this stuff, you've made all these discoveries, but you've not actually had any impact at all in terms of patient care. So we set about looking at gene therapy and using adeno-associated viruses to try and have an impact on disease. At the same time, I was involved in clinical trials, a lot of clinical trials that came and went and had no efficacy whatsoever. But one of my first patients who was involved in the antisense trial targeting SOD1 when the drug was called Tofersen. And I saw the first 4 patients treated in the world and saw that after 3 months, SOD1 was knocked down. And more importantly, neurofilaments, a marker for neuronal degeneration, was also reduced. There was just 4 patients that were treated and 4 patients that were given placebo. And I knew that this was an efficacious therapy. And then, of course, this has gone on, and now these drugs are licensed. Sadly, it's not a miraculous cure. I've got a couple of patients who've had a really remarkable response in which the disease has been arrested. But many patients do progress, and if you've got very aggressive disease, then the impact is still pretty modest. So, this was the door opening a crack to show that we could have an impact in this disease with a genetic-focused therapy. And so that was pretty exciting. I've been involved in 2 C9 trials that didn't show any efficacy, even though the biomarkers were showing promise. And we've been involved in the IONIS trial on FUS as well. So around this time was the most remarkable evidence of efficacy using adeno-associated virus, which was to supplement the gene was missing in spinal muscular atrophy. You've heard about spinal muscular atrophy from Tom yesterday, but basically it's a survival motor neuron gene is either missing a key element, was mutated so that these patients are homozygously deficient. And giving a virus in the first 6 to 9 months of life that carries the gene for the missing survival motor neuron gene turns these children into pretty normal, healthy children. These children all die or on ventilators by the age of 2, and some of them now at school, going to ballet and living a pretty normal life, which is absolutely remarkable. And this was proven in the first 18 patients. 17 out of the 18 had a really dramatic response. So I felt this really laid down the gauntlet, to bring therapies to patients and to either supplement or silence genes. And so we spent the next 6 or 7 years trying to do this. But there are problems. These were infants, and in infancy, the blood-brain barrier is a bit loose. And 2 billion years of evolution have led all humans to have a pretty good effective blood-brain barrier to prevent viruses going across. And people forget that when you put something into the cerebrospinal fluid, it doesn't always get into the brain. There's a membrane around the brain, the spinal cord, called the peel membrane. It's 4 to 6 cells thick. And big structures like a virus doesn't necessarily cross there. And of course, then curing mice are easy, but the human brain is an awful lot larger. So the idea that you could treat something in a mouse with a virus to then translate immediately to humans is a bit unrealistic. And so looking at how these viruses are delivered, you see a really low rate of transduction of neurons in the brain and spinal cord. And indeed, this is work in macaques showing that we see these tiny pockets of transduced cells in the brain, and this is the work of Jim Wilson. And when actually Siddharthan and I were there when we first presented this work, and I said, oh, those are Virchow-Robin spaces. And he said, what are they? And I said, well, that's where the arteries are penetrating into the brain substance. And most of the cells around it are astrocytes. And he said, oh, yes, most of those are astrocytes. So the percentage of neurons that are transduced in the brain is very, very low. But what you do see is in the lumbar spinal cord, quite a high transduction, and that's because there are holes in the peel membrane. We don't know why they're there, but they are there, and it means virus does get in. But in the cervical cord, it's 0 to 2% of transduction, and those neurons innervate your arms and your diaphragm. Most of our patients want to breathe and to be able to use their hands. So this was not a viable approach. And the other problem was that many of these monkeys, and indeed people, we discovered later, developed degeneration of the dorsal root ganglia, because the dorsal root ganglia are floating around in the CSF, and they take up a lot of the virus, so it's toxic. This is an example of this. This is a patient of Bob Brown's, and they gave a gene silencing microRNA in an AAV intrathecally into the spinal fluid. And this chap had a very aggressive form of disease. Now, when it starts on one limb, it usually crosses over into the other limb. So this disease started in his left leg, and that's the upright triangles. And you see power here measured in the y-axis, and there's no power in that leg, so it's completely paralyzed. And so what normally happens is the right leg would start to decline, and you can see it does start to decline. And at this point here, with a dotted line, they gave him the injection of the microRNA AAV, and it continued to decline for a while, but after about 15 weeks, the leg started to improve, and it stayed strong throughout his life, even though the arms became weak and his breathing was affected, and he died after 60 weeks. This leg was preserved, and the motor neurons were preserved. But SOD1, this is the Western blot for SOD1 down the bottom here, was almost undetectable. So the virus and the microRNA worked. It got into the lumbar spinal cord through the holes in the peel membrane and suppressed the toxic protein being made and rescued those neurons, but it didn't get to the rest of the nervous system. The other terrible thing that happened to this chap was he had very severe sensory neuropathy. He was actually admitted to hospital, and he had pain for 20 weeks. So, a mixed message, but if you can get the virus to the right place, to the right cells, you can turn this disease around. That was the message. Anyway, so when I was thinking about doing this, I was, approaching my 60s, and I thought, old dog, new tricks, is this a good idea? I could be having a quite comfortable life, writing the occasional grant and writing a paper or two. But we decided to step out and do it, but we wanted to do it differently. And everybody else was thinking about putting virus into the cerebrospinal fluid, and I didn't think that was a good idea because I didn't think it would get into the brain sufficiently at sufficient levels. And I quite like this Larson cartoon. Everybody else was, walking in V formation, but we were going to try and fly. It's a bold claim. And we decided to start off with a secreted protein, progranulin. And Siddharthan was there when I raised the progranulin story to Jim Wilson, who then claims it's his idea. And he didn't have ever heard of it. He didn't know what FDD was. But we said this was a good target because it was a secreted protein. And the idea was very simple, and that we would ask the thalamus, which is one of the most connected parts of the brain, to be the factory of progranulin by giving it the virus, and then it would be transported up axons and secreted at synapses. Well, some of those bits of the story had been published, but nobody had ever done it before. And so we tried to sell this idea to investors to be able to build a company to be able to afford to make the virus, to be able to afford to do the experiments. So with Yeonbok Lee and Do-Young Lee, who have experience in AAV, technologies, we designed this particular construct, which is has a variety of clever elements that every single viral capsid is able to make more progranulin than its original, and that it was under a very specific promoter that's neuronal. So we asked the neurons to be making it because they were connected throughout the cortex and could do it, could do the delivery. We didn't want it expressed in other cells that were dividing because it's known to be able to accelerate tumors, and high levels of serum progranulin are associated with poor prognosis for most solid and blood-based cancers. So a lot of safety issues there. We first of all treated mice that were knocked out for progranulin, and you can see that there's lipofuscinosis, this red stain here, and when we gave the therapy in the thalamus, we could see that disappeared. The late manifestations in these animals is they develop an inflammatory reaction, and this is CD68 positive microglia, and again, we were able to suppress that signal pretty effectively. In fact, even very low doses were able to do this. So we're excited about that. And the no self-respecting New Zealand scientist doesn't do an experiment in sheep. And so we worked with Stephen Gill. We put the virus into the thalamus, or he put the virus into the thalamus. And this is the sheep receiving its infusion of the virus quite happily. And this is the cannulas that we put into the sheep's brain, which delivered the virus. And we could monitor this by putting gadolinium into the mix as well to see the distribution. And of course, all good things come to an end, including this video. And we compared the thalamus that had been injected on one side with the progranulin construct and looked at the progranulin levels as opposed to saline, and we could see a very big difference in terms of the distribution of progranulin. So that was pretty exciting. And we could see that it was expressed not just in neurons, but it was actually also taken up by astrocytes and even microglia. So we managed to get delivery to about 80% of cells. We then went on with a lot more investment to do a safety experiment and dosing experiment in non-human primates. This is the non-human primate data. This is the thalamus. You can see very, very high levels of progranulin in the thalamus. Fortunately, we didn't see a toxicity signal after 6 months. That was very promising. And we could see distribution of all these different doses across all of the cortex. And we could see levels in the CSF rising. And of course, we were pleased to see that the levels in the serum didn't rise because we didn't want high levels of serum progranulin. Anyway, we're now in clinical trials. We've treated 8 patients so far. We do this very carefully, very slowly, with an infusion into the thalamus. Under MRI guidance, we use gadolinium to see where the infusion is going. And this is now an internationally open trial, including the UK, finally, after 5 years. Anyway, this is one of the first patients that was treated in March 2024. And you can see the catheter track here and the gadolinium that's gone in. And we do this bilaterally. So there are 2 injections to 2 sides. The procedure takes about 8 hours. So it's a non-trivial procedure, but we have shown that it's relatively safe. We've not had any serious adverse events. Anyway, so frontotemporal dementia, we're able to supplement. We're excited about this opportunity to try and use the thalamic connections, I call it a thalamic sprinkler, to deliver our product around the brain. But my passion is ALS. There's a lot of toxic genes that make toxic proteins. We want to suppress that. And so we built a platform for silencing, and we particularly targeted MAPT, SOD1, and ataxin-2. And so we've named this VMIX, which is one-time gene silencing. We're using blood-brain barrier crossing capsids, and we are able to regulate expression in certain cells by using promoters and enhancers. This is where it gets a bit commercial. So this is all the work now done in AvioBio. The original VMIX platform was developed in my lab at King's. But it's now being developed and expanded through AvioBio. So what happens here is it's very much like antisense. It's processed by the brain. It's a normal physiological process. It involves a nuclear step in which it's taken from pre-microRNA, exported and cleaved again until it's into a double-stranded form. It's taken up by the RISC complex. It splits the passenger and hopefully excludes that, and the guide stays in the RISC complex and able to bind to the target mRNA and suppress it. We did a lot of design features to try and improve the specificity of this, enhance guide selection and depress passenger selection. And we then looked at sort of plug and play manipulation of the guide so that we could have a platform that was stable, but we could choose a different gene target and just change about 120 base pairs to have a completely different target. So that took 6 years. And again, this is the work of Romain Joubert and Euan MacDonald, and we have this microRNA expression cassette, which we think is pretty cool. Anyway, so we've done lots of experiments in mice. I was cynical about treating mice, but we are able to knock down SOD1 in mice. We've got more work going on in terms of looking at the clinical impact of that. But this is just a single microRNA, and we're able to show in between 1 to 10 vector genomes per cell. So even if you get just one genome in that cell, we're able to sufficiently knock it down, probably by about 70%, which should have important therapeutic impact. When you think about tofersen having efficacy, they show that CSF levels drop by only 30%. So we're hoping that we can do better than that. And the construct we've chosen in the end has got very high guide to passenger ratio. So the passenger here is orange and the guide is green. So that's very positive because if the passenger is included in your risk complex, you can get off-target effects. So the other gene we've been targeting is ataxin-2. There's a long story as why people are interested in ataxin-2, but the most striking disease-modifying effect is in TDP-43 mice in which you've knocked out at the genomic level ataxin-2. Some of those mice, instead of living only 30 days, live 400 days. And again, we've got a construct with a single microRNA, which is able to knock it down in the 1 to 10 vector genomes per cell ratio, around about 70%. So we think that's important. Okay, so now we've got an even better microRNA, which is a triple stem microRNA. Again, we've managed to generate this, stabilize the platform, and show that it can be manufactured. And we see even greater knockdown. In this case, it's MAPT tau as a target. Anyway, so we're very pleased with ourselves about this particular construct. It is plug and play. If you give us a gene target, we can come up with something that will knock it down within about 6 months and bring that to preclinical studies in mice. And of course, isn't just on the CNS. If you put it into a vector that'll target other tissues, it could be used elsewhere. So that's one of the advantages. So Aviato Bio has not a capsid company. So we've been working on the payload for the last nearly 10 years, but we wanted to get it across the blood-brain barrier. And so we've been liaising with companies who've been making these new capsids. And one of them is this company called Apertura, and they've modified the capsid so that it expresses an epitope that binds to the human transferrin receptor. And that's great because that transferrin receptor is being used to traffic transferrin-adapted proteins, peptides, RNAi, and small molecules into the brain, and you get transcytosine delivery of your product from the bloodstream. And what we're wanting to do is, instead of doing an 8-hour infusion under MRI guidance, just give somebody a single injection. And of course, if you're transducing neurons and you die mostly with the neurons you're born with, if you stably transduce them, then that's a therapy for life, one-time therapy. So we have another vector that we're working with a Japanese company with, which also has fantastic delivery across the blood-brain barrier, at least in macaques and in mice. But we don't know what the receptor is. And so we're probably going to be doing some work on the background of that to find out what that receptor is. This is the second blood-brain barrier crossing capsid. This is a non-human primate, it's a macaque. And you can see all the green cortical cells that are positive. It's about 80% of all neurons that are positive, and it lights up the spinal cord, particularly in the motor neurons as well. So this is a very exciting opportunity, if we can do it safely, to deliver our vector across the blood-brain barrier with gene supplement and gene silencing platforms. And this is the Apertura capsid, which binds to the transferrin receptor. We're now using a mouse that has been knocked in for the human transferrin receptor. And that's exciting work. And here you can see one such mouse, which is expressing in scarlet, and the non-transgenic mouse doesn't get across the blood-brain barrier at all. Also very good in the spinal cord, particularly in the motor neurons. Okay, so this is what I think we've reached to the stage so far. We're getting exciting developments from antisense, giving us a clue that we can have an impact on these disorders by manipulating gene expression. We've shown that we can get our proteins around the brain if they're secreted by asking well-connected parts of the nervous system to deliver that. But actually deliver across the whole nervous system is now feasible through blood-brain barrier crossing capsids. And of course, giving an intravenous injection could be done pretty much in any hospital. And hopefully, we can do that at doses that are safe and efficacious. So the most important thing whenever you start something up is to employ really clever people who are able to do the job much better than you. And I'm very lucky to do that. Lisa Deschamps, I should say, was the person who brought Avexis into Novartis and then did the global launch for the spinal muscular atrophy therapies, or Gensma. And then everybody else on the panel there have previously worked in gene therapy programs. And my co-founders, Jan Lee and Euan Lee. These are our, what I call blue chip investors. I won't go through and name them all, but been very lucky to survive this long in biotech, which is credit to all the team, and hopefully to go forward. And this is some of the team that are in my lab at King's, because we still do all the small animal work for them, and then lots of people who've come through. So firstly, to say that the motivation for this is all about trying to change the future for therapies for patients with ALS. And that's a promise I made to my patients in Bury St Edmunds 30-something years ago. Lots of people have funded the work over the years, particularly UK DRI, the Doddy Foundation, Van Gies Foundation, and of course Aviato Bio continue to fund the work. And I've got lots of collaborators, some of whom are in the room, but mostly to thank the patients who've contributed their samples and helped drive this research forward. Thank you.
Thanks, Chris. That was an inspiring talk. Questions? Oh, Seth.
Great. Thanks, Chris. Terrifically interesting talk. Vast diversity of neurons in the brain with different transcriptional profiles. You're targeting a message that, through your microRNA approach. Have you ever looked to see what the effects of these tools are in different classes and types of neurons by using single-cell approaches?
So we haven't done single-cell, and that's a really good point. We have done total microRNA-seq and RNA-seq, and surprisingly little deviation. So at the doses we're using, we often see very little major disruption or change in the abundance of these proteins. Now, what we were worried about is that we might interfere with normal microRNA biogenesis, but we're not seeing that, at least at whole tissue level. But I think single-cell seq is probably going to be the way to go.
Okay. I'm really struck by the fact that I think you showed like 80% of neurons, or you get knocked down in 80% of neurons. So you really must be hitting a very broad set of classes of neurons, and in all of those having some sort of measurable
it's extraordinary.
effect. So it sounds like that's a very useful tool for people who just want to knock down genes in the mammalian central nervous system.
Sure. Yeah, if you don't want to do it by CRISPR and knock it out generally here.
Any other questions? Over there.
Thank you for a very interesting talk. I was curious with the emergence of new model tools such as induced pluripotent stem cells based on human biology, have you considered within Avideo Bio using those models instead of mice?
Yes, we are using those models. You still need an in vivo approach in some sense. We do a lot of our early screening in iPS-derived neurons. I think going to organoids is a little bit bigger step because of the complexity of cells we're targeting. But yeah, we do a lot of our early work now on iPS neurons.
Thank you.
Chris, can I ask you one question? I mean, this is fantastic, and we all know that about 80% of this ALS cases are sporadic. What are your thoughts? And there are new tools emerging with TDP-reg and everything. And do you mind giving your insight on whether that could be adaptable with your platform, the AAV therapies and stuff?
So what was
the TDP regs, the new ones, the regulatory TDP?
Yeah. So I think the knocking down genes that are mutated and toxic is a great start, but we need to think about sporadic disease. And in fact, the ATAXN2 program is really about trying to reduce TDP-43 misaggregation. And what's interesting about the original papers from Aaron Gittler's group is that they show TDP-43 in the knockout mice is actually, there's very little accumulating. And so we see that as a potential therapy. But I think we also need to think about removing misfolded TDP-43. And I think that's where an antibody's trim approaches to try and enhance clearance is going to be important. I think both of those things. The nice thing about AAV is we can probably package both the silencing and a supplement to aid clearance as well. So I think that's an exciting area.
Yeah, fantastic. One last question? Any burning question? If not, again, thank you, Chris, for your fantastic talk. So our next speaker is Andrea Serio. He's from King's DRI, and many of you will know Andrea started his PhD in Edinburgh as an EMC member as well, and then he moved with an engineering background, I remember, Andrea. And then he moved to Molly Stevens' lab in Imperial and developed these human stem cell models into much more asking fundamental questions in axon length and dynamics around the axonal, looking at the axonal dynamics. And is back in King's DRI and working on ALS disease modeling. Andrea.
Right, thank you very much for the introduction. Well, first of all, it's very special to be here. Thanks to Siddharthan and everybody else to make this day possible, and thanks to the EMC for making the larger this possible. As Bhuvaneish said, we heard a lot about how Euan's legacy has changed lives, but also changed scientific careers. Certainly has influenced and shaped mine quite significantly. As Bhuvaneish said, I was the first EMC Edinburgh-based PhD student at Siddharthan's lab when Siddharthan wasn't in Edinburgh yet. In fact, the lab when I started was David's story and a bunch of boxes, and that was quite an experience. For this talk, I sort of dug out somehow the very first image of the very first presentation I've ever given as an EMC student. And it was something on the line of this, where we're trying to connect how to get to meaningful therapies for ALS through better understanding and getting better understanding through, well, better modeling. As we heard yesterday, one of the things that Siddharthan was bringing into the expertise of the EMC when he joined was the human-based stem cell models. And that was very much exactly what my project was focused on, bringing about better modeling to understand better how the disease comes about, and perhaps also use it, as Chris said, for trying and testing new therapies at the early stages. And so, as we've seen on the last couple of days, there's a plethora of different ways that we can assemble together to model MNDs and ALS and FTD. There's a number of animal models that are fantastic to recapitulate certain aspects of the disease, and there's a number of cell culture models that actually can give us a lot of lateral ways of movement to get mechanism nailed down. And the in vitro system is great for control and reproducibility. The in vivo system is great for the physiological relevance. And my very first project in science, my scientific career here at the EMC, was to sort of move the needle from the in vitro work slightly more towards the physiological relevance by introducing complex models that incorporated more cell types. My interest was neuronal glial interactions, and I made protocols to generate astrocytes and neurons and assemble them together and try to understand how the crosstalk between these two cells was relevant for TDP-43 proteinopathies and ALS in particular. And that was a very successful PhD and very much directed my future scientific career, because in the intervening 12 years now, I have continued to essentially do the same thing and try to move the in vitro part of our modeling towards delivering more complex, next generation human-specific models for neurodegeneration, and thanks to my original upbringing at EMC, specifically for MNDs and ALS in particular. So my lab currently is a combination of engineers and neurobiologists, and the activities around our research are around three main topics, and I'm going to show you a little bit of what we are doing at the moment, starting of course with what is probably the most prevalent activity in our lab, which is centered around engineering motor neurons for the purpose of generating better models of ALS. Now, one of the very first observations that I had in the lab when I joined, and that sort of influenced the very first project that we did in my lab as an independent career, was that although we could very efficiently generate lower motor neurons and other forms of spinal cord motor neurons in vitro, and it worked very well, they were also remarkably different from the in vivo counterpart that we were trying to model. More striking to me, having an upbringing as an engineer and a biotechnologist, was that their actual dimensions, their engineering principle behind them was remarkably different. While we were trying to model something that can be up to a meter long in vivo, we were very happy to have something that was a couple of 100 microns in our dish. And that always was a little bit strange to me, and one of the questions that remained over the course of my PhD and postdoc was, does this profound difference in scale matter when it comes to modeling how a neuron, well, lives and dies in motor neuron disease? So the very first question that we wanted to answer in my group was whether the question of length in vitro models, whether a neuron has different length, matters to its actual function and its homeostatic pressure. And this is not a question that we could actually answer very easily because there was no platform at the time that could allow us to take the length as a single variable and analyze it. So we had to spend about 5 years in developing a new microfluidic microfabrication-based system that allowed us to create a controllable in vitro systematic length system. We called it LAM because we don't have a lot of fantasy for names. It stands for long axons for motor neurons. And although it has a remarkable name, it was a very cool platform because it allows us to generate this spinal cord organoid from iPSC cultures that are entirely open to the culture medium. They're not enclosed in any microfluidic systems, and they are placed on a micropattern surface of microgrooves, and that allows them to essentially generate an array of oriented axons that can be grown for microns, millimeters, and several centimeters even. And so for the first time, we had access to a system that allowed us to effectively control the axonal length of our cells in vitro. And with it, we had actually discovered something quite important, that axonal length in vitro matters quite a lot. We discovered that for motor neurons and now for other subtypes of neurons in vitro, at a certain threshold length, there are specific biophysical switches that are reenacted and change and remodel quite significantly the mechanism that control the homeostasis of the axon itself. Particularly striking for us was the fact that at the length of about 3 to 5 millimeters in length, in these motor neurons, we were observing drastic changes in the mitochondria, drastic changes in the local translation, and more importantly, drastic changes into specific RNA-binding proteins, TDP43, FAS, and many of the other RNA-binding proteins that are associated with ALS. So for us, a lot of the things that we would have wanted to look at in terms of phenotyping these cells for motor neuron disease were drastically different when looked at in a long axon than in a shorter one. And so recapitulating, the engineering characteristics of the neurons seem to have a profound effect on the type of phenotypes we were able to actually observe in them. We published this as a general mechanism for wild-type cells in 2022, and then immediately we set about to understand how could we use this discovery and platform to better model MND. Particularly exciting for me was the opportunity to model two aspects of the pathophysiology of MND, which is the axis of time and space. Because while we have obviously a temporal axis where we have to understand what are the early stages of event of the pathology compared to the late mechanism that enacts towards the end of the life cycle of a neuron, we also have a spatial axis. Some of these mechanisms will take place in the cytoplasm towards the nucleus or in the nucleus itself, but most others will actually probably start at the very end of the axon in the axonal synaptic compartment. And the possibility with our platform to model these two aspects was quite exciting. And nowadays we have a large body of work and a number of projects that are all focused on understanding the links between metabolic and transcriptional dynamics in these axons. And we have a number of activities that are focused on using these insights for better understanding ALS in particular. So a couple of examples that I want to give you today. It's how we used the LAM platform itself in its current iteration to try to phenotype a large battery of ALS relevant lines. So the first thing that we did was we generated an array of engineered iPSC lines based on the same COLF 2.1J golden state background that were carrying several mutations on proteins associated with familiar cases of ALS. Here you have an example from FAS and TDP, and we started making LAM arrays in both short and long form. And the first thing that we checked was that we were able to actually get for both our wild types and our ALS lines the same axonal length of 1 centimeter or more without having any deficits due to problems with either the knocking of the mutations or the mutations themselves. And that was very exciting because it meant that we could actually do this experiment, but also even more exciting when we started looking at some of the phenotypes that you would normally want to study in these iPSC-based models, we started noticing that the long axons were remarkably different from the short one. For example, one of the first things that we wanted to look at is the mislocalization of the mutant proteins. When you have mutations in some of these RNA-binding proteins, such as TDP and FAS, one of the first noticeable features is that they tend to mislocalize from mostly nuclear to cytoplasmic or even axonal. Well, as you can see here, this is an example of TDP43 densitometry across the axon in all of our lines, TDP, FAS, and wild type, and the equivalent experiment for FAS, comparing short and long axons. And as you will appreciate, the phenotypes that we observe with the short one and the long one are very different, particularly with TDP43, as you can see here. We can observe that in TDP43 mutant axons, we can have a noticeable mislocalization of TDP43 itself when it's mutated, which is entirely not present or not visible when the axon is much shorter. And a very equivalent effect is with FAS in FAS mutant axons when they are long compared to the equivalent system when they are short. In other words, this is another example of how recapitulating the length of the axon is bringing out a much more potent and visible phenotype that we would otherwise have in much shorter axons. And I should say that this is also, we call them short, but as you can appreciate here, they are still almost 5 millimeters in length, which is way longer than they would be in any normal cell culture, for example, in a 96-well plate. Another example is we found a number of differences, again, in the mitochondrial phenotypes that we could observe. The mitochondria of both TDP43 mutant axons in the long configuration and FAS are very different from their wild type counterparts. And again, we have a phenotype that is completely opposite when we look in long axons and short axons. Particularly in the long axons, we tend to observe this varying mitochondrial length that indicates that in these mutant axons at the long distal components, we have changes into the fusion and fission dynamics that actually regulate the mitochondrial shuttling and the dynamics themselves. And just show some more preliminary data, we recently also being able to partner up with a mitochondrial geneticist, a collaborator of ours in Italy, and send them different fractions of the axon from which they were able to extract the mitochondria, extract the mitochondrial genome at the different components of the axon, and check for the number of genomic variants present in this mitochondria. And as you can see here, comparing the soma, the proximal, the medial, and the distal fractions of our axons across 1 centimeter, and comparing the TDP and FAS, for example, to the wild type, we observe that, especially in the axon, a higher amount of single nucleotide variants for mitochondrial genomic DNA. And that's actually quite indicative of the fact that we have these complete changes in the mitochondrial shape itself. Now, this is part of a large body of work that we hope to get published later on this year. We think it's going to show that recapitulating the axonal dynamics and shape actually has a large payoff and get a better model and a better set of phenotypes in our in vitro systems. However, this still is a very descriptive set of phenotyping experiments where we are just effectively trying to understand how the different mutations are changing the balance of specific phenotypes that we know are targets for the disease. One of the other things that we wanted to do is turn our platform into a bit more of a blinded discovery system where we could, in an unbiased way, understand what's going on in these very long axons across a variety of different molecular classes in order to do some more targeted discovery for potential new therapies. So one of the things that we spent some time doing is making sure that our platform was very scalable. So compared to other systems that perform similarly like microfluidic devices, we were able to expand our platform to include several hundred millions of cells for each device, up to now 150 millions. And then we were able to also eliminate any barrier that we would have to extract the material from this platform. So there's no plastic or PDMS or silicone enclosing the cells themselves. And we spent some time optimizing the salami slicing of our axons so that we could effectively grow these arrays of long axons and then extract thin slices of them up to a couple of millimeters in length, from which then we could start to do some molecular analysis. And I hope you had time yesterday to check one of our talented senior postdoc here at the posters, and where she has presented essentially this idea of performing axonal multi-omic analysis to map the entire biomolecular structure of the axon, doing lipids, protein, RNA, and for the somatic part, chromatin analysis as well. And we are now, thanks to the support of an MNDA grant, trying to assemble a data space where we can combine all of these different omics modalities for wild type, TDP, FAS, and VCP mutants, and try to compare different subtypes of neurons and different length of axons, so that we can sort of start answering questions about what are the early stages changes we can see in the pathology, and what are some of the aspects of selective vulnerability, for example, when we compare axons that belong to cortical neurons or to motor neurons or to other subtypes of neurons that are less affected in the disease. We also had a chance, and I want to mention this very briefly, to start to expand, thanks to other collaborators within King's, to more specific intervention when it comes to axonal homeostasis. Particularly, I want to mention one of the other posters that was here yesterday, that is our PhD student, Sophia Fredin. She is working with Jamin Sheridan, who also was named in Chris's talk, and she is trying to optimize a ATP set of visualization sensors that will allow us with an imaging-based platform to essentially observe ATP, which is the base currency for metabolism within the cells, and it's going to be very relevant in the axon as well. And she's trying to perform what we call spatial dynamic metabolomic by imaging in the axon. So in other words, she's going to be able to map the ATP domains along these 1 centimeter long axons and correlate them with all of the other mechanisms that we are finding when we are analyzing the lipids, proteins, and RNA dynamics across them. And if you haven't had a chance to talk to her or a poster, she is over there. She's a very tall Swedish PhD student. She's going to stand up above the crowd, literally. This is all well and good. It's very exciting. I'm very excited about the engineering of these cells. But as we've seen yesterday morning, one cell is just not going to cut it. A lot of the very relevant phenotypes and common features that we can observe in animal models and in the patients have to do with circuit level abnormalities, with functional problems in the network dynamics in ALS and FTD. And so we tried also to do an effort and developed our platform so they could incorporate to a certain level the network and circuit dynamics. Particularly, I've been lucky enough that we had a PhD student in our lab, Pacharporn Suklai, that just published her paper, and she was able to take all of the platforms that I've just shown you to make single arrays of axons, essentially treat them as Lego blocks in a larger construction, and develop a protocol to assemble them in an easy way without the need of any specialized equipment into a large format combinatorial circuit that can be effectively changed and adapted to any configuration one might want, up to several centimeters squares in size. And the way it works is very easy. It uses the same microfabricated device and microgroove systems, and it's able to assemble them in effectively any configuration, which means that it's now possible to take an arbitrary network design, such as this one, where you have two separate nodes of neurons that are supposed to connect in a specific way. And this might be to model, for example, different layer connectivity in the cortex, or in this case, for example, a specific simplified connectivity of upper to lower motor neurons. And once we have sketched that out in PowerPoint, as I've shown here, we can actually recreate it out of iPSC-derived, patient-derived cells carrying the mutations. And as you can see here, we can develop it into these large format, 1 centimeter square circuits that connect over time at the macroscopic scale, but they are also able to be investigated down to the single axon to single dendrite synaptic putative contact points that form at specific points within our platform that we decide in advance. In other words, we are adding an element of predictability to a circuit that we can build from scratch in the lab with patient cells. And as you can appreciate here, one of the things that we were able to do is, because we are deciding in advance which axons are connecting to which dendrites in which position, we know in advance where the synapses are going to form. And as you can see here, we can predict the large number of contacts are going to happen at a specific location. And then when we go and check, in those particular locations, we have a biasing up to 2 times as many active synapses while we maintain the same number of neurons. In other words, we are increasing the yield of synapses in a specific part of our circuit, which then we can probe with the same mechanism that we are using for the axon. For example, in this case, we are able to disassemble the complex circuit into its constituents and separate, for example, the synaptic containing region from the cell bodies and the axon. And one thing that, for example, is not possible to do with any other microfluidic system, we were able to do proteomics and now also phosphoproteomics in the future. For example, here you're seeing a proteomic analysis of the extraction from the different parts of this circuit. And as you can see here, we are able to separate quite clearly this T-junction bottom, which is where we drive the number of synapses up. And then when we actually see how this particular part of the circuit is separated from all of the other components, as you can see here, it's mostly because we have enriched for a number of synaptic proteins. And we're very excited because now, thanks to the support of an M&D accelerator grant in collaboration with Mark David Roop's lab at the UK DRI at King's, we are using this very platform to test prospective new therapies to maintain synaptic connectivity in FTD and ALS using this iPSC-derived circuit model. And we're very excited about it. We are also trying to improve our motor neuron connectivity, because obviously neuron-to-neuron connectivity is all well and good when we're talking about FTD and when we're talking about the upper motor circuits. But because we are interested in ALS, we also want to do something about the muscle-to-neuron connectivity. We had to spend quite a long time trying to decide what was the best way to try to implement neuromuscular junctions in our system. And in the past 4 years or so, we've been lucky enough to be involved in a large European-funded consortium called MAGIC, which is a gene therapy consortium focused on muscular dystrophies and trying to solve certain congenital pediatric muscular dystrophies with gene therapy. And our job into this consortium was to generate an ecosystem of bioengineered products that would allow us to create mini muscles, large format bioengineered muscle construct from the very same iPSC cells that we were using for the motor neuron studies. And I'm not going to go into very many of the engineering details here, but the way it works is that we can generate this hanging construct, as you can see here, in between an hydrogel. We can culture iPSC-derived myoblasts that are under tension because of the 2 pillars, and over time they differentiate into titin-positive contractile fibers that we can then put in contact with other cultures, or we can use for testing AAV vectors in this case, or more importantly for us, that we can match with our iPSC-derived motor neurons and try to start making really functioning neuromuscular cultures here. I don't know if the video shows that very well, but what you're observing here, it's a blob of motor neurons that we are coupled with our muscle engineered construct, and the contractions that you're seeing and the calcium spikes that you're seeing are as a consequence of us stimulating the neurons directly on the construct, which in other words is a nice way of saying that this is a neuron to muscle communication happening within one of these constructs. And we are now we are now in the process of generating all of the muscle from the TDP43, FAS, and BCP lines and matching them with the corresponding motor neurons in these constructs. I hope I convinced you that there is some quite interesting, exciting new things that we can do combining engineering and biology for these platforms. In the last minute or so, I just want to talk about the unsung heroes of this, which is the part of the lab that actually focuses mostly on the engineering and the hardware around these platforms, which is very essential for what we do. And particularly, I want to mention one of the things that we are focusing on right now. As I mentioned at the beginning, I showed you during the talk, one of the things that we are trying to do is to bring about a next generation of complex human-specific models to try to understand better ALS and other diseases in vitro. And that is fantastic because it allows us to increase the complexity and increase the dynamic behavior of the system. But this also comes with the downsides of we have to admit that once the complexity increases, we tend to lose control, we tend to increase the variability, we tend to increase the workload, and it becomes much more complex to work with the systems. One of the things that we have spent about a decade now doing is trying to understand how can we introduce these complex models, but at the same time introduce also the tools to make sure that the research can continue. And we have now developed an entire instrument platform that is a robotic bioprinting, liquid handling, cell incubation microscope system that effectively acts as a biofactory for example, brain-derived organoids that is able to generate them, culture them, analyze them, and sample them in a completely automated way, and brings about what we think is going to be the self-driving experiment for advanced biology. And just to give you a very quick example of what we're able to do with this, this is a culture of cortical brain organoids that we generated with the machine. Every one of these little green balls, it's a collection of about 500,000 neurons, and what the machine is able to do is generate them, culture them for about 28 days there, survey their electrical activity, and as you can see here, isolate the ones that have a certain threshold of interconnectivity and therefore maturation, and then we can decide which specific one we are interested in for example, single-cell RNA-seq or further analysis, and the machine effectively goes and automatically picks up one single organoid from this complex culture while maintaining the culture alive. And if you're interested, we have a preprint now out that is currently in review in Nature, and we even have a different preprint where one of our collaborators has already used it for a pipeline, and we hope to spin it out into a company later on this year. Now, I will leave you with probably the most important slides, which is the people that have actually done all the work, that I've been very fortunate to work with, particularly Kathleen and Sophia are here if you want to talk to the real scientists behind this, but I've been very lucky to be able to work with some incredibly talented young scientists, and I would like to thank all of our funders and our collaborators and you for your attention, and I'll be able to take any questions. Thank you very much.
Thanks, Andrea. That was truly phenomenal in pushing the boundaries of stem cell disease modeling. Thank you. A couple of questions. Maria? And then Matt.
Thank you for the fantastic talk, Andrea.
Thank you.
Just two questions, brief
Sure.
ones. Have you been able to correlate the elongated mitochondria with mitochondrial function, for example, by measuring mitochondrial respiration?
So it's currently ongoing, so we don't have any definitive data yet, but what we're being able to correlate some of the morphology phenotypes is with the next NGS of their actual genomes. We are currently running through the functional correlation for the mitochondria.
Great. And my last question is, do you think the elongated mitochondria is a genotype-driven effect, or have you seen it in stressed but otherwise healthy neurons as well?
So we observed in the original paper that the lengthening of the axon has an effect on the morphology of the mitochondria, but it's opposite to the one that we observe when we have actually the mutations. So we think that the increase in single copy variants of mitochondrial genomes we see, it's an effect rather than a cause, because these are not patient-derived lines. They are all COLF 2.1J with a knock-in mutation, and we think it has to do with probably the shuttling dynamics that mitochondria have in the axon.
Matt.
Hiya.
Hi.
Thank you very much. Very cool talk. Have
Thank
you
you.
thought about, or is there any value, do you think, in creating vertical cultures? Because as long as axonal length is a really important part, but also the direction in which they have to project, and and having to do, for example, transport processes that might work against gravity, can actually add an additional stress that's implicated in some aspects of motor neuron vulnerability in ALS.
Believe it or not, for entirely unrelated reasons, we have tried to put these cultures vertically, horizontally, counterclockwise and clockwise. It's a funny story, but we haven't done any specific studies on that. It's a very good idea. We should probably look a little bit more into that. Yeah. Thank you.
Sarah? Last question.
Hi. Oh, Hi, I'm Sarah McGlasson from the DRI here in Edinburgh. This is a bit of self-interest, really, but and I'd love to speak to you some more. But have you thought about vascularizing any of your microfluidic systems? Maybe look at neurovascular unit coupling?
Yeah, we've thought about it. It's not our... If I had to be hated by my lab for adding one bit of complexity, I would like to, the hill that I would like to die on, it's the myelination first, and then the vascularization. We don't seem to have the same problem that organoids have, because although they are three-dimensional cultures, they are well below 500 to 600 microns on each side to the center, and so we don't seem to have a lot of problems at the moment for that. But it's definitely one of the on the list for the future.
I'd love to maybe speak to you more about vascular microfluidics.
One last question from Jamie.
Hi, thank you. That was a great talk.
Thank you.
I wonder, how well do you think you're capturing the dendritic architecture?
Ooh. We haven't done much on the dendritic architecture because they branch and it's complex, and we need to sort of have a micro-patterning sort of shape that allows us to look at that. We have a few projects that we started in the lab a few years ago in terms of trying to array the dendritic tree in the same way, but we haven't actually done much progress. It's not something we have invested enough time. It's a very good point.
All right. Thank you, Andrea. So the next session is, we've got 4 emerging leaders from Euan MacDonald Centre. So the first speaker is Matt Broadhead. So he did his PhD in Edinburgh with Seth Brand, and then he moved to St. Andrews working with Gareth Miles' lab, and he'll be talking about synaptic diversity in motor neuron disease.
Thank you. Thank you very much, Bhuvaneish. Thank you very much for inviting me to be here. Yeah, my name's Matthew Broadhead. I'm a researcher at St. Andrews University. I did my PhD here many, many years ago. I'm really excited to give this talk. The overall theme of my research is on synaptic diversity and vulnerability in ALS. The truth is that the real story of this talk is very much about a serendipitous finding. It's about curiosity-driven research from there and the implications that that can now have for ALS. And I do feel like there's a real space for this within the EMC research community. So a day in the life of what I do, I'm a microscopist. I use a lot of high-resolution and super-resolution microscopes to visualize the nervous system in ways that make it look like this. Lots of weird green, blue, pink dots. Many of these structures all represent synapses. These are the small chemical communication junctions between our neurons. They are very small, they're very complicated, they are incredibly diverse throughout the nervous system, and we know that synapses are highly vulnerable structures in various neurodegenerative disorders, including ALS. So for the past few years, a lot of my research has been focused on finding vulnerable synapses with a view towards trying to understand how we could preserve them. Some examples of this, 3 years ago, we looked at tripartite synapses. These are synapses that receive specialized communication connections with astrocytes, the supportive cells. And we found that tripartite synapses in a mouse model of ALS and in human post-mortem tissue were significantly more vulnerable to being lost than the uncontacted synapses. From there, we used super-resolution microscopy to study the nanoscale organization of TDP43 inside synaptic compartments and investigated to what degree that inferred some degree of synaptic vulnerability. And most recently, we've been looking at lots of different types of synapses in the spinal cord in a more relatively novel mouse model of sporadic ALS. This is just a sort of a flavor of the kind of case studies of the things that I've been sort of working on, but it was during the course of all of this I stumbled across something. So it was a day where we had about 4 years ago, we had a new microscope in the department, and I wanted to go and test it out. I took over a piece of spinal cord that for me is a bit of a test sample. It was a spinal cord from a mouse model that was actually generated by Seth Grant here in Edinburgh. It expresses a postsynaptic protein, PSD95, fused to a green fluorescent protein, GFP. So we can just light up synapses throughout the nervous system very, very easily without having to do any labeling. And I took a big 3D volumetric image in the spinal cord near where my motor neurons might be. Well, not mine, the mouse's. And it took me a very long time looking at this image and just gawping at the data until I saw something weird in the top left-hand corner, just about there. It was a huge, large, sort of continuous matrix of this PSD95 labeling that was filled with lots of perforations. The structure is about 5 to 10 times bigger than your average synapse. There's some little puncture down there at the bottom for reference. That's about the size of a typical postsynaptic density. So this thing's big. Of course, this is just N of 1 out of about 4,000 random structures in this entire image. But from that point, I just kept an eye out for, do I see these structures ever again? More and more, and I looked back at old data and even then collected new data, we found that these structures were real synapses. They're opposed to presynaptic boutons that express VGLUT1, and we often found them on the cell bodies of motor neurons, either labeled with CHAT or the large alpha-type motor neuron marker of MMP9. So from that point, we thought it's very plausible that this synapse could be part of the monosynaptic reflex. This is basically our knee-jerk reflex, right? It's our simplest microcircuit that we kind of have. So you'd think we'd know as much as we can know about this circuit. So we did some, we gained some mouse tissue from a collaborator at Glasgow University to light up the sensory neurons of this circuit, and then with immunolabeling, we're able to sort of visualize whether these weird multi-perforated synapses were part of this circuit. And to cut the story a little bit short, yes, they were part of this circuit. In fact, they were almost exclusively a part of this circuit. Like 95% of them were opposed to parvalbumin positive and VGLUT1 boutons. But what this data shows on the left-hand side is that there's a huge amount of diversity in this simple circuit. You get some very, very small, simple-looking synapses. Most of the synapses look quite complex. They have multiple different PSD sites. About 5 to 10% of them showed this multi-perforated morphology. At this point, I felt like we needed a better name for them. I'd been calling them multi-perforated postsynaptic densities, which is a mouthful. Various names kept getting thrown about within the lab, and a colleague at the time, Simon Sharples, kept saying, we should call them the honeycombs. Honeycomb, that's a cool name. I thought it was a bit of a daft name at the time. But we were doing some super resolution microscopy to look more in detail at the nanostructure of this synapse, and what we see is that PSD95, in many cases, it's not homogeneously organized within a synapse. It forms hotspots, if you like, as you see from these kind of like heat maps. These are distinct nanoclusters that align with presynaptic release sites. There are about 5 to 6 nanoclusters of PSD95 that form around each perforation in a sort of hexagonal-like ring. And actually, there are about, on average, 6 perforations per synapse. So actually, okay, I said to Simon, okay, fine, you know what, you win. We'll call it the honeycomb synapse. And I think it's quite a fitting name for it. Now, all the while, this is just a side curiosity that had caught my interest. And I kept wanting to find reasons to leave it, but it kept finding its way back. But at the time, I was also interested in ALS and trying to understand what is happening to different types of synapses in ALS. And we just kind of come to the end of some work where we had used lots of different types of synaptic markers to look at lots of different types of synapses in the spinal cord of a mouse model of ALS that shows inducible TDP43 pathology and motor deficits. And we could show that certain types of synapses were more affected than others. All the while doing this, I kind of had this thought of, what if it was possible to visualize all of the different types of synaptic inputs to a single motor neuron at the same time, essentially, so that we could gain a much deeper insight into what is happening for each individual neuron and its entire catalog of synaptic inputs. This is a challenge, but I've been trying to achieve this using a multiplexed imaging approach, whereby we label a motor neuron and then sequentially and iteratively label different types of synaptic markers and then image them and wash them away and label for different rounds of synaptic markers. So we can build up that much greater catalog of the different types of synaptic inputs that come from different types of neurons, and we can gain a much richer data set as to what is happening on a single neuron basis. So I performed this quite recently on some healthy control mice and some of these TDP43 mutant mice that show quite a severe motor deficit, and sort of try to catalog and look at different types of these synapses. And what this graph shows is the number of synaptic structures per perimeter of the motor neuron, normalized in each case to their control levels, and we see quite a very specific and selective case of synaptic loss. We are losing both the VGLUT1 puncture and the PSD95 counterparts of these 1A afferent sensory synapses, perhaps a lot more so than any of the other synapses that we examined. Now we know, though, now that these synapses are diverse. So what's happening to, for example, the honeycomb synapses from within this population? Well, in the control animals, about 25% in this case of all of the VGLUT1 synapses onto these motor neurons showed this honeycomb postsynaptic morphology. I've not just ignored to put the data there for the ALS group. There were no synapses. There were no honeycomb synapses at all. It was entirely abolished. They weren't just reduced. They weren't just changed a bit. They just weren't there. So I've not done the stats on that, but I promise you it's probably quite significant. It's one of the few cases where I don't have to go to SPSS. So I think from this, I think it highlights, hopefully, two key take-home messages. One is the focus on a scientific point of view on synaptic vulnerability and synaptic diversity. We've heard a lot about, certainly in the first day, about the vulnerability of certain types of neurons. And what this evidence sort of shows is that actually, if you look at synapses, you can get such considerable degree of diversity within a very simple circuit, from the same sensory neurons to the same motor neurons, and you've got different types of synapses, and some of those will be more vulnerable than others. So I do think there's some value in rephrasing our focus onto which types of synapses may be more vulnerable than others, as opposed to necessarily which types of neurons. The other key take-home message is this, and I do think this is really important for fellow early career researchers and students, is to just take time to look at data. Like, I had missed this. I've been looking at these synapses in this sort of area for many years until I just gawked at that data for long enough. What really frustrates me is that I've got an image from 2016 when I first started my postdoc with Gareth, and I can see one of these honeycomb synapses, and I missed it. And so I do think there's some real value in just taking the time to look at raw data. And I do appreciate that we all in this room want to develop a cure. You know, we're working against the clock. We want to one day deliver a cure that will cure this disease tomorrow. But I do think we need to provide more time for researchers to just sit and look at data, and otherwise we might just miss something. And with that, I'd like to thank the various people that I've had the chance to work with and collaborate with. We have some incredibly fantastic students at St. Andrews that are so engaged from the undergraduate level to the postgraduate level. But I do think it's really important. I really want to thank my supervisor, Gareth Miles, who has always supported my somewhat wild and wacky ideas and just allowed me a lot of free reign and freedom. And it's been incredibly valuable for me. And thank you very much to the EMC community for allowing me to be here to talk. Thank you.
Thank you, Matt. Thank you, Matt. That was fantastic. Learned a new thing, honeycomb synapses.
There we go.
Questions? A couple of... over there.
Thank you for a wonderful presentation. So I thought a honeycomb-shaped synapse is very interesting biologically. So do you observe those synapses generally, widely across brain, or only in specific neuronal subtype or specific brain region?
I do wonder to what degree they may exist elsewhere. If you look in the literature, the idea of a perforated postsynaptic density is not entirely novel. People have shown this through electron microscopy in the cortex. I have never seen any or many structures describing it in this way, or even therefore defining that nanostructural level, that sort of nanostructural organization at this level. But this is the first time we've seen this in the spinal cord. But I'd be very keen to see any other examples where people have found weird and wonderful synapses, and maybe there are some common threads of synaptic logic into how these synapses are built in this way.
Thank you.
Sorry, lovely talk, thank you. Since we touched upon inhibition excitation yesterday already, but rather with focus on the cortex, I was wondering, or let's say I was surprised to see in your data that you don't seem to find any changes in this particular mouse model on inhibitory synapses onto motor neurons, which, for instance, has been shown for the SOT mouse model, right? So you seem to primarily see this very selective input or deficit on the sensory input, which is probably reminiscent to some degree of SMA, isn't it?
Yes, so this VGLUT1 pathway is very well known in the MND field, particularly with SMA. You're right, it was a bit of a surprise not to see those changes in the inhibitory synapses. I suppose one caveat to that is that, firstly, we are using a relatively pan-inhibitory synapse set of markers. Yeah. And again, if anything, what this highlights is that there is undoubtedly further degrees of diversity, and perhaps if you were able to really sub-define out distinct circuits, you might start to see where cases of pathology arise, and perhaps when you look at a more global synaptic markers, they might be missed. So if anything, what my data is trying to say, I'm not trying to say, no, there are no inhibitory synaptic changes. It's just that when I do it this way, I don't see anything, and that also doesn't suggest that there could still be functional changes at these synapses as well.
I absolutely believe that there is heterogeneity also, of course, with the patients, right? And I think we might need better tools to really detect which patient would benefit from what kind of interference or procedure, And so if you have data that argues against a loss of inhibitory input, I think that's very important, right?
Yes, one of my thoughts is that, like, as much as we don't like the mouse models for lots of reasons, if we were able to refine our protocols to be able to do these kinds of assays across multiple different mouse models, it wouldn't be that hard. It's just getting all of the tissue together and running it. And then we might start to see the common threads, or we might start to see those differences, and that might infer something about patient stratification.
Thank you.
Thank you.
In the interest of time, we should just move on. Thanks, Matt.
Thank you.
Our next speaker is Helena Chaytow. She's a postdoc with Tom Gillingwater's lab, and she's also a ECR champion for Euan MacDonald Centre. And she'll be talking about neuromuscular junction and how to target them in animal models. Helena.
Great. Thank you so much, Bhuvaneish. So I am going to talk to you today about our work looking at the immune system and the neuromuscular junction. Before I start, I just wanted to take a minute to talk about my small contribution to the EMC network. Every 6 months we have our research afternoons, and I set up a postdoc networking session for the mornings of those research days, inviting all postdocs from any of the labs in the EMC to come and talk together. Postdocs are at a really important stage of our research careers. We're developing new ideas, we're writing grants, we're establishing ourselves as independent researchers. And the EMC provides an amazing opportunity for us to discuss those ideas, consider collaborating with people from different skill sets, and ultimately carry out stronger research. We've, in these networking sessions, we have shared with each other our current research. We've had troubleshooting sessions, and I've organised for funders to come and talk to us about funding opportunities available to us as early career researchers. Ultimately, we aim to form strong collaborations within the EMC network at the lab bench to harness the amazing breadth and depth of research that's undertaken in the EMC, which you can appreciate from the range of expertise just at this one networking session. I apologise for the colour scheme. I made the mistake of using the Microsoft Smart Art for that, and I can't change it now. But anyway, I like the honeycomb. Apparently that's the theme of the session. And if you can't read it, I promise you there's a breadth and depth. I only had the pleasure of meeting Euan once at one of the public research evenings at the Anne Rowling Clinic, but here I hope we continue building on his legacy of collaboration in M&D research. And if these postdoc networking sessions are new ideas to you and you haven't heard of them before, watch out for emails from Rebecca and Pam or myself before the next research afternoon and come along and collaborate. Okay, back to the science. So, as you are all aware in this room, the neuromuscular junction is a key point of pathology in ALS progression. And here we have, Here we have an image of a human neuromuscular junction. We have a healthy, fully innervated neuromuscular junction with a motor neuron in green, and a neuromuscular junction from an ALS patient that has completely lost that innervation. And these denervated NMJs are replicated in our mouse models. So this behind me is an image from our aggressive mouse model, the neuronal-specific TDP43 overexpression mouse model. And you can see in cyan, we have the motor neuron growing down into the muscle, covering the end plates in a healthy mouse. But in the diseased mouse, you have fully denervated and partially innervated neuromuscular junctions. So this is a point of pathology. We also know that the immune system plays a role in ALS development. There are multiple factors that drive immune dysfunction in ALS. There are mutations that impact the immune system. There are protein aggregates that might activate the immune system, environmental stresses or disease modifiers that might change how the immune system works. Ultimately, evidence from ALS patients indicates that there's both excessive inflammation and an inefficient immune response. So blocking the inflammatory immune response may be neuroprotective. And we wanted to see whether this immune dysfunction actually has an impact at the NMJ itself. So in collaboration with Sonia Alfonso Martin's lab in San Sebastian, we looked in the skeletal muscle of ALS patients. And we found infiltration of CD45 positive cells into the skeletal muscle, specifically in ALS patients. So you can see the cell infiltration here, and that's quantified in this graph. And at least some of these cells seem to be of a macrophage lineage. They were CD68 positive. So we see this, these symptoms of immune cell infiltration in ALS patients, but do we replicate those in our ALS models? And as has been mentioned several times over the last couple of days, there's no perfect mouse model for ALS. So we looked at 3 of them. In our lab, we work a lot with this fast-progressing, early-onset, neuronal-specific TDP43 overexpression mouse model, and we see a very significant increase in CD45 cells in the skeletal muscle. If we look at a fast-progressing but adult-onset form of ALS mouse model, the A315T model, we still see this significant increase in CD45 positive cells. And then even if we look at a very slow-progressing adult-onset form, so these mice only show very mild motor weakness at 12 months of age, if we look at that time point, we still see this CD45 positive cell infiltration into the skeletal muscle. So these immune cells are found in skeletal muscle across mouse models and in ALS patients. And we looked to see if they were kind of interacting with the NMJ at all. And interestingly, we found significantly more CD45 positive cells around the innervation zone than in the non-innervation zone. So if you're not a muscle biologist, you might not be aware that there are bits of your muscle that don't have NMJs in them, and the NMJs cluster in innervation zones. And it seems that the immune cells are attracted to those innervation zones. So then we asked what could be causing this attraction, and so we did a proteomic analysis of inflammatory markers in the skeletal muscle of our ALS model, and we found a range of inflammatory markers were dysregulated in the muscle, but particularly we found an increased expression of the chemokine CCL2. Immunohistochemistry showed that CCL2 expression was actually localized to the NMJ. So there are cells found at the neuromuscular junction that are expressing CCL2. And this CCL2 expression is an early phenotype. So in our mouse model, the CCL2 expressing cells are seen at the neuromuscular junction before these mice develop any motor weakness at all. And this CCL2 expression continues throughout the disease progression of the mouse model. So then that's the CCL2, that's the ligand side of the attraction. So where is the CCR2? Well, indeed, the infiltrating immune cells into the skeletal muscle are positive for CCR2. So we've shown that there's co-localization between the CD45 infiltrating cells and the CCR2 here. And again, this is an early pathology, so at early symptomatic stages, you see a significant increase in the CCR2 positive cells in our mouse model. And then we went back to our ALS patient tissue and again found CCR2 positive cells in the skeletal muscle. So this CCL2, CCR2 kind of dysregulation is consistent in both mouse models and in ALS patients. So we looked to see if we could block it. Using an intramuscular injection of CCL2 specific neutralizing antibody or a pan-IgG antibody, we injected the immune modulation into the gastrocnemius muscle of our mouse model, and then we could compare that injected muscle with the contralateral uninjected control. So this is the contralateral control here. You can see that a lot of the neuromuscular junctions are denervated. And if the muscle is treated with either the IgG antibodies or the CCL2 antibodies, you get restored innervation of the end plates. This is that data quantified here. And then we looked at other markers, other parameters of the motor neuron and found that there was an increase in nerve terminal area, in axon diameter, and in percentage overlap. So we have improved presynaptic parameters, for example, axon diameter. And then finally, we also looked to see if we were having any impact on the infiltrating cells themselves, staining for CD45 and CCR2, and we found that there was a significant reduction in the infiltrating cells in the injected muscle and almost a complete absence of the CCR2 positive cells following the treatment. So, in conclusion, we have immune cell infiltration into skeletal muscle as a key pathology in both our ALS models and in patients. This seems to, at least in part, be driven by the CCL2-CCR2 axis, and it can be blocked by neutralizing antibodies, improving the NMJ innervation, improving motor neuron parameters, and reducing the immune cell infiltration. And my future work hopes to develop this new therapeutic strategy into a more translational approach that can move towards the clinic. Finally, I would like to thank particularly Bernat McGrady from Szeged. He is an incredibly talented clinical fellow who worked with us here in Edinburgh for 12 months, but is a long-standing collaboration. Thank you to Tom Gillingwater for supervising and mentoring my work. Thank you to all of our funders, everyone else in the lab, and thank you for listening.
Thank you, Helena. A couple of questions. Richard? Oh, Brian Dickey, sorry.
Is Richard
Brian,
going first?
No, I thought it was Richard. But Brian, go ahead.
I'll take that as a compliment. It's just a question looking at the human tissue, because I'm always keen to bring this to human disease. Have you or could you look at primary lateral sclerosis, first of all, because you don't get the muscle wasting occurring? And secondly, maybe look at traditionally resistant motor neuron populations and therefore muscles, such as the ocular motor muscles or onus nuclei, which control sphincter function.
Yeah, absolutely. I think that would be a fascinating control to see if this immune cell infiltration is indeed playing a role in the denervation, then we wouldn't expect to find the cell infiltration at the NMJs of the extraocular muscles, for example. It would be a matter of sampling, I think. So if I had access to those samples, then I can perform the analysis, absolutely.
Richard.
I knew and feared that Richard would have a question.
It's neuromuscular junction, so of course you'd expect it. Have you had a chance to look at the chronocyte population of cells at NMJs, which are CD34 positive?
That is something that I want to do next, definitely. We wonder whether that might be the cell population that is expressing the CCL2 at the neuromuscular junction itself. We've looked at whether it's the terminal Schwann cells, and it doesn't seem to have full overlap with S100 staining. So we would want to look at the chronocytes as well, definitely.
Guy.
Hi, Guy.
Yeah, fascinating work. Being an electrophysiologist, I want to know whether your neuromuscular junctions work. They look better, but are they any better functionally? Have you got any plans to look at that?
Good question. Yeah, this was just an acute experiment at the moment. We were just looking at changes in the anatomy of the NMJ. But going forward, we want to look at whether this treatment actually has an impact on the electrophysiology properties of the muscle in overall grip strength, that kind of thing. Yeah, definitely on our list of things to do.
Yeah. Can I ask you one question? What are your thoughts on why CCL2 is upregulated in the muscle? How does it, because you've checked it in various mouse models, say some of them where pathology is expressed only in the neuron, some of them in whole systemic.
Yeah. Yeah, not sure. Not sure why CCL2 is upregulated. Like you say, it seems to be a consistent phenotype across whether the transgene is expressed in muscle tissue. Sometimes it is, sometimes it isn't in those mouse models. And in the ALS patients that we took tissue from, they had different genetic backgrounds, and some of them didn't have any known genetic mutations. So the cause of this CCL2 upregulation is not known. Yeah.
Okay. Thank you.
Thank you very much.
Thank you, Helena. So our next speaker is Caroline McHutchison. So she's again Edinburgh graduate, worked with Sharon Abrams, and now lecturer at Stirling University. And she'll be talking about cognitive behaviors in MND.
Well, thank you very much for the introduction and for the opportunity to speak today. So I'm going to be talking about something a little bit different. So my background's in neuropsychology, and my research really focuses on understanding when cognitive and behavioural symptoms first begin and how they develop over time in MND. And today I'm going to be focusing on one aspect of my work where we're looking to characterise mild cognitive impairment and mild behavioural impairment in pre-symptomatic MND. So first of all, I'm going to just briefly introduce the Miami framework, which has been proposed to describe the development of both MND and FTD. So when we start first with motor neuron disease, it's likely that before someone receives a diagnosis of MND, that they pass through a pre-symptomatic stage of the disease. And so this might first start with a clinically silent phase where we can only observe evidence of the disease based on biomarkers. But it might also then be followed by a prodromal stage where we start to see these mild signs and symptoms of MND, but they're not clinically significant enough to meet the diagnostic criteria for MND. Based on this framework and the clinical and biological and genetic overlap between MND and FTD, the framework also includes this frontal temporal axis. And this aims to capture mild cognitive and behavioural symptoms that are associated with frontal temporal dementia and also clinically manifest frontal temporal dementia. And given this framework, an individual may present at any point in either of these axes at any given point in time. When someone has a diagnosis of MND, we know the types of cognitive and behavioural impairments that they experience. These have been well characterised in the literature. I'm just going to mention them briefly because Professor Sharon Abrahams is going to talk about these in more detail in the next session. But the types of cognitive difficulties that we see tend to centre around what we call executive functions. So these are difficulties with things such as planning, making decisions, perhaps holding bits of information in someone's mind and being able to manipulate it. But we sometimes also see difficulties with what we call social cognition. So the ability to read and understand emotions, thoughts, and feelings in other people. In addition to executive functioning, we sometimes see difficulties with language as well. So maybe difficulties naming things, with spelling or comprehension. And when people have MND, we have the strong criteria which has been established to identify people who have these mild cognitive and behavioural impairments. And we refer to this as ALSCI and ALSBI. But there has been growing interest in whether these mild cognitive and behavioural impairments are present in the pre-symptomatic stage of MND. And as a result, there are a lot of different frameworks that have been proposed, both from the perspectives of ALS or MND, but also from the perspectives of the FTD community as well. So the specific study that I'm going to talk about today, we aim to examine the frequency or prevalence of mild cognitive and behavioural impairment in pre-symptomatic motor neuron disease. And to do this, we wanted to take these frameworks that we had identified, compare them, established a refined criteria to identify mild cognitive and behavioural impairment, and then explore the prevalence and types of characteristics that we see in this prodromal stage of MND. So to do this, we use data from the ongoing pre-symptomatic familial ALS study, also known as PREFALS. So this is a study that is ongoing. It's being conducted by researchers at the University of Miami. And it recruits individuals who carry a genetic risk of MND or FTD, but they don't have any clinical signs of the disease at the time of enrolment. They receive study visits every 1 to 3 years, and these are very intensive, comprehensive, multimodal assessments. And it includes comprehensive neuropsychological testing, which was introduced in 2011, but also the brief assessment, the Edinburgh Cognitive and Behavioural ALS Screen, or ECAS, which was introduced in 2014. All of this data has been reviewed by myself, a research neuropsychologist, but also as part of a clinical consensus team where needed. So in total, we have 216 individuals who have neuropsychological data, and collectively they have data at 807 time points. As we can see, 188 of these individuals do have a genetic risk of MND and FTD, and these are predominantly C9 and SOD1 carriers. We have one smaller group of gene-negative participants. These are people who come from a family where there is a history of motor neuron disease, but they themselves don't carry a known genetic risk. So one of the first things that we did was we took the pre-symptomatic or the prodromal frameworks that we identified in the literature and we applied them to some data from the pre-FAL study. Due to the data that we had available and some difficulty operationalizing some of the criteria that had been published, we were unfortunately only able to apply 2 of these criteria. We did also apply the strong criteria because we recognize that it's important to have consistency in classification across the different stages of the disease. But what we found was that the rates or the estimates of MCI and MBI really varied depending on the framework that was applied. So for cognitive impairment, it ranged from 12 to 35%, and behavioral impairment ranged from 17 to 50%. The criteria in the blue in the middle is from the FTD community. This combines both cognition and behavior together, and we can see that the prevalence ranges from 18 to 45%, depending on whether we use the criteria which is specifically developed for gene carriers or not. But the main takeaway message from this is that across all 3 of these frameworks, only 38% of visits were consistently classified depending on the framework that was used. So this meant 62% of visits had different classifications depending on the framework that was used. And when we look at each of these frameworks, we see that they differ in a number of important ways that are likely contributing to this variation in the estimates of MCI and MBI. And these include how they measure decline, how do they measure cognitive and behavioral decline, what domains are they focusing on? Are they focusing on the ALS-specific domains such as executive functioning and language functioning, or do they also include other domains like memory and visual spatial skills? And lastly, do they allow classification based on a single time point, or do they require evidence of decline based on objective measures? And so we explored each of these characteristics iteratively to develop a refined MCI criteria, which is a whole nother presentation in itself. So I'm just gonna move on to the prevalence, but I'm happy to sort of answer any questions about that later. So when we applied this criteria to all participants who had neuropsychological data in the pre-FAL study, we found that 16% of gene carriers met the criteria for MCI at any given time point. This is compared to 4% of people who were gene negative participants. I realize that says gene positive, but the group in the magenta are the gene negatives. And when we look at this group of people who have a genetic risk and they meet the criteria for MCI, the type of profile that we see of neuropsychological impairments is most often multi-domain. So that means it includes difficulties with tests of executive functioning and also tests of language. This happened in around 68% of individuals. The remaining 32 had a single domain cognitive impairment, and this was most often captured by difficulties with what we call the letter fluency test, which is the ability to generate words that meet a certain condition, which we know to be very sensitive in MND. We also looked at these individuals to see if there was any different based on the type of genotype that they had. And what we found was that there was no significant difference in terms of the prevalence of MCI across different genotypes. And one of the last things that we did was look to see how MCI status changes over time. So this is a subset of individuals from the pre-FAL study, mainly for illustrative purposes. And this is a state transition plot. So I'm just going to walk you through for anyone who's not familiar with these plots. So each row here represents a single individual, and each of the dots represents a specific study visit. We can see that the lines in dark blue, the navy color, these are visits where they've been classified as having MCI. The green are where they are unimpaired, and the light blue is this middle category where we see some evidence of deficits or decline, but they're not severe enough to be classified as MCI. This may be due to impairment on one test only, or perhaps the presence of a confound such as depression. But really what this plot is showing is that there's a lot of fluctuation and variation in MCI status over time. And we know from the Alzheimer's disease world that this is also observed in a prodromal stage, but often it can be an indicator of an increased future risk of development of FTD, or cognitive and behavioral impairment. So I think over time it's going to be really interesting to see how these fluctuations pan out. So I've really focused on the cognitive impairment here. This has been our primary focus, and now we're hoping to move on to the behavioral symptoms. But just to wrap up, I'd love to just acknowledge all of my collaborators, but also mentors, Sharon, but also Michael Benatar and Joanne Wu from the University of Miami, as well as all of the participants and the amazing team that collect the data on this study over in Miami, but also my funders and the EMC for just all of the opportunities to share my work with all of these researchers. Thank you.
Thank you, Caroline. Quick question? Alyssa?
Hi, thank you. That was a really interesting talk. I was interested that when you stratified amongst genotypes or genetic mutations, you didn't see any specificity towards, say, C9 with the FTD spectrum.
Yeah.
I was curious if you further separate those C9 patients, do you see any stratification between who then develops FTD versus who develops ALS, or are we not far enough along to know?
Not quite far enough along to know that, but I think that's really interesting, yeah, because we maybe would have expected to see a significantly higher prevalence within the C9 carriers. There is some growing research which sort of indicates that perhaps SOD1 do also experience difficulties with cognition. But yeah, I think that's a really interesting thing to look at in the future. Yeah,
thank you.
Can I ask you one? Yeah. You mentioned in one of your slides there's some biomarker analysis is being done. Can you comment on what sort of analysis, and are you planning to correlate with them?
Yeah, so the pre-file study, the data collection for each individual takes about 2 days. So there are a lot of different neurofilaments being collected, brain scans. And yeah, so the next stage is sort of to look at, do people with MCI or MBI also have differences in certain biomarkers? And I think that's a really important step, because we really are focusing here on people who have a genetic risk of the disease. And I think tying in those biomarkers is what's going to make it applicable in the sporadic population as well.
Any further questions? If not, thank you once again, Caroline. We've got our last speaker, Maria Stavrou. So Maria is a consultant neurologist, and she worked with Siddharthan and I for a good long time now. And she's got she's going to talk about axonal neuronal, astrocyte neuronal crosstalk, and particularly around the mitochondrial transport. Thank you, Maria.
Thank you, Bhuvaneish. Good morning, everyone. I just want to start by saying that I'm deeply honored to be here today celebrating Euan. I had the privilege of meeting him a couple of times, and I have to say that our conversations have actually stayed with me. And his calm strength and optimism were truly remarkable. So what I can say is that indeed we're going to continue being inspired and guided by his vision. So as Bhuvaneish said, I'm a newly appointed consultant neurologist in Edinburgh, and I co-lead the Motor Neuron Disease Service with Richard Davenport, Savan Kapal, and Siddharthan. Alongside my clinical work, I'm a senior research fellow at the university. And my research basically builds on my PhD, which was independently funded by the Medical Research Council, and centers on understanding how astrocytes regulate axonal transport in ALS. This is a grossly unexplored field, so I look forward to sharing my work with you and getting your insights. So having heard so many outstanding talks on motor neuron disease, ranging from its devastating clinical presentation to the pathophysiology, I know very well that this group does not need any primers. But in ALS, fundamentals do matter, and therefore recognizing that sporadic and familial ALS are clinically and pathologically indistinguishable highlights the value of studying a single genetic disorder to unravel this complex condition. And this is the exact reason I chose to study the C9orf72 repeat expansion, because it is by far the biggest genetic contributor of ALS and frontotemporal dementia. Its key features can be faithfully reproduced in an iPS-derived model. And thirdly, and importantly, it mirrors sporadic ALS, including TDP43 pathology. So in ALS, axonal transport seems to be exactly where cell biology, genetics, and vulnerability intersect. We know that motor neurons have meter-long axons, which are heavily dependent on the ATP, which are heavily dependent on the transfer and maintenance of mitochondria. We also know that there are several genetic mutations that hit this system, and this can range from microtubule networks to impairment of adaptor complexes, as well as RNA cytoskeletal links. Nevertheless, all of them converge to one main outcome, which is impaired axonal mitochondrial transport. Using multiple patient-derived lines carrying the C9orf72 mutation, our lab and others have demonstrated that the C9 mutant motor neurons have impaired axonal transport, shown in pink here, and this is accompanied by reduced mitochondrial respiration. But crucially, both of these pathological phenotypes are reversed in their gene-edited controls. Additionally, when we overexpress PGC1 alpha, which is a master regulator of mitochondrial metabolism and biogenesis in the C9 mutant neurons, we're able to rescue this axonal transport deficit. So together, these data suggest a mechanistic link between impaired axonal transport and bioenergetic dysfunction in C9 ALS pathophysiology. All of the studies until now, nevertheless, have looked at the intraneuronal consequences of these genetic mutations on axonal trafficking. What about astrocytes, though? We already know that they are master regulators of the central nervous system metabolism, but could they modulate axonal transport in ALS? And this is an unmapped area which I set out to address. So for this work, I generated highly enriched astrocyte cultures using the 3 independent iPS-derived lines, carrying the C9orf72 mutation along with their gene-edited controls that were generated using the CRISPR-Cas9 technology. These astrocytes have more than 90% GFAP and S100 beta, which are astrocyte markers, and importantly, they are functionally competent. In other words, they can take up glutamate and they can propagate calcium waves. Crucially, these C9 mutant astrocytes recapitulate key pathological features of C9 ALS, including RNA foci and dipeptide repeats, all of which features are abolished in their gene-edited controls. So my next question was, how does the C9orf72 mutation affect the metabolism of the astrocytes? And to address this, I used the Seahorse assay and measured glycolysis and mitochondrial bioenergetics. And what we see here is that the mutant astrocytes, shown in red, have diminished basal, ATP-linked, and maximal respiration, as well as impaired glycolytic capacity, although I'm not showing the data here today. And all of these deficits are reversed in their gene-edited controls, and we're able to replicate this across the 3 independent pairs. Having demonstrated this metabolic deficiency in the C9 mutant astrocytes, I subsequently wanted to investigate the non-cell autonomous consequences on motor neurons. To do this, I set up physical co-cultures. I used astrocytes across the 3 independent patient control pairs and co-cultured them with motor neurons from one C9 iPS line and its gene-edited control. For mitochondrial transport in the neurons, I measured 2 well-established features: the percentage of motile mitochondria and the average speed of those motile mitochondria. So I started by looking at the control gene-edited neurons in isolation, and as expected, the results recapitulated what was previously seen in the lab. But when those were co-cultured with the C9 mutant astrocytes, there was a clear reduction in mitochondrial transport. And importantly, upon physical contact with the gene-edited astrocytes, this deficit was reversed. And I was again able to replicate this pattern across the 3 pairs. Next, I turn to velocity. When these control neurons were in physical contact with the C9 mutant astrocytes, in orange again, there was a reduction in the average speed of motor mitochondria. And this is a notable finding because when we just looked at the C9 mutant motor neurons in isolation, we did not identify this genotype-driven deficit. So it looks like that this phenotype is driven by astrocytes, pointing to a non-cell autonomous mechanism. And these videos essentially illustrate what we just discussed. I did try very hard to find a witty, memorable analogy for you, but clearly I failed. But I guess we can confidently say that the first one probably represents what British Rail would look like. Very slow and sometimes absent movement. And this is what involves the mutant astrocytes. So I then proceeded to the next question. How do control astrocytes affect these diseased motor neurons? Again, I looked at the motor neurons in isolation, and I recapitulated the findings that our labs and others had previously shown, which was reduced mitochondrial transport. Upon co-culture with the C9 mutant astrocytes, although there was no reduction in the percentage mitochondrial motility, probably due to a floor effect, what we see is that there is a reduction in the speed of the motor mitochondria. And again, both phenotypes are rescued upon physical contact with the gene-edited healthy astrocytes. And these findings are consistent across the 3 pairs. So overall, we can conclude that in C9 ALS, the axonal transport phenotype is dictated by the astrocyte genotype and not by the genotype of the neuron. Given the bioenergetic deficit in the astrocytes I talked about at the start, the next obvious question was, could this be driving the neuronal transport defect? And to test this, we overexpressed PGC1 alpha in the C9 mutant astrocytes and repeated the co-cultures. If this is the case, then the neuronal transport would rescue itself. And this is exactly what happened. So again, I'm showing the motor neurons in isolation as before, followed by the co-cultures with the mutant astrocytes. But crucially, what we see here is that when the C9 mutant astrocytes overexpress PGC1 alpha, the mitochondrial motility is restored. And this is clearly shown in the chymographs here. This is my penultimate slide, so bear with me. So just to start understanding the mechanistic basis of how these C9 astrocytes control axonal transport, I decided to measure mitochondrial respiration in motor neurons co-cultured with astrocytes. And I know that many of you will start wondering now, how are you able to differentiate between a neuronal and astrocytic respiration? I'm more than happy to answer that in the Q&A section. But in summary, what we saw is that when the mutant or the isogenic neurons are in co-cultures with the mutant astrocytes, what is shown in olive and orange here, there is a significant reduction in mitochondrial respiration, and this is improved upon physical contact with the gene-edited healthy astrocytes. And we validated these findings by measuring steady-state mitochondrial membrane potential with mitotracker CMX-ROS, and we saw the same pattern. So the C9 astrocytes do modulate mitochondrial bioenergetics in motor neurons. So in summary, I hope I have been able to convince you today that in C9 ALS, the axonal transport phenotype is determined by the genotype of the astrocyte and not the neuron, that the astrocyte genotype directly influences mitochondrial bioenergetics in motor neurons, and that by boosting astrocyte bioenergetics to overexpressing PGC1 alpha is sufficient to rescue the axonal transport deficits. And all of this uncovers a novel role of astrocytes in regulating axonal mitochondrial transport. But as you can imagine, this raises many more questions than answers, which I hope I'll be able to address in the future, including how astrocytes mechanistically do this Is it metabolic coupling? Is it mitochondrial transfer? Is it both? Do they affect the transport of other organelles? And crucially, is this relevant in sporadic ALS? I'd like to stop here and thank everyone for listening. I'd like to thank my supervisors, my lab colleagues and collaborators, and of course the Euan MacDonald Centre, because it has created a truly unique community for clinicians, for scientists, and people with motor neuron disease to work side by side and achieve progress. Thank you so much.
Thank you, Maria. Seth.
Fascinating talk. Can you say a few quick words about what are the mechanisms that transport mitochondria, and are they in any way unique to mitochondria? What else is transported by those mechanisms?
Well,
including
yes.
proteins and presynaptic
of course.
molecules.
This is a very interesting question. So we have anterograde and retrograde transport of the mitochondria. For the anterograde transport, we have the kinin. For the retrograde transport, we have the dynein. And we now know that the amount of ATP used by these two different motors is actually different. And those motors can also transport other organelles, like endosomes, for example, or mRNAs or peroxisomes. And in fact, I just completed the analysis for endosomal tracking. And what we see is that in fact, there is no difference across conditions. And this probably has to do with the energy that these organelles use. We know that the mitochondria are more dependent on the TCA cycle, and therefore they are impacted by the metabolic deficiency in astrocytes, but the endosomes are more dependent on glycolysis. So yeah, we'll see, much more to come.
Sabine?
Full talk, thank you. I must have missed this, but you said multiple times in physical contact with astrocytes.
Yes.
So have you tested whether that is, or whether factors that might be secreted might also play a role? In other words, have you just used, what's been done
Actually,
10 years ago, or just
that was my very first experiment. So astrocyte conditioned medium,
Yes.
but unfortunately we didn't see any effect of that. So it looks like this effect it requires this physical contact between the astrocytes and the neurons.
So it could be a sort of loss of function,
Indeed.
right? A
Yes.
physiological function that is needed to take.
Indeed.
Okay, thank you.
Gareth?
For some reason, all the questions are over here. Really nice talk. I'm just wondering, how specific do you feel the effects of the mitochondrial or the bioenergetics rather of astrocytes is to axonal transport? I wonder whether when you correct that, this is going to actually have a whole raft of other benefits to the function of the motor neurons. And maybe if I can add a second part, I guess you're saying contact's important. So if we think in vivo, where are astrocytes contacting motor neuron axons? Yeah. That's a limited part of the axon.
I know.
So that's kind of an interesting element as well. Sorry, two questions.
Yeah, thank you. So starting from the last question, we do know that astrocytes are confined to the central nervous system. But at the same time, those spinal astrocytes are what provide metabolic and trophic support to the somata of spinal neurons before they extend peripherally. So probably what happens centrally affects the axons distally. But this is where the value of complementary models comes in, and I really want to validate these findings in an appropriate mouse model. And the first question, which I don't remember, sorry.
About going beyond axonal transport, I mean, this could be really beneficial to lots of aspects of motor neuron function. What's your thoughts about how specific what you're looking at is for transport versus other functions, as you say, maybe in the cell soma?
Yeah, there is some work from Bhuvanesh's lab, actually, that when they managed to boost axonal transport, they were able to find improved neuromuscular junction projection. So I guess there are many other outcomes we can look into. Yeah.
Peter?
Yeah. Your last slide mentioned transfer of mitochondria. Have you got to the point of looking at that yet, whether there's any movement of mitochondria from astrocytes to neurons?
Yeah, we do have some preliminary data on control astrocytes and neurons, and we found that about 30% of, there were about 30% of astrocytic mitochondria on neurons, but this is just preliminary data.
Yeah, sure, thank you.
Yeah, thank you.
One last question.
Thank you very much for the talk. I guess I was kind of wondering if you think there's a specific time point where this axonal transport could be going wrong. Do you think it's rather something that is occurring during the development of a neuron? Or do you think that if you were to culture neurons separately and then later add sort of the healthy versus diseased astrocytes, whether you would see also these acute effects that inhibit the axonal transport in the way you described it?
I mean, we know that the iPS models we use have developmental features. We know that. But at the same time, astrocyte neuronal metabolic coupling and mitochondrial metabolism are fundamental processes, so we do not think these are stage dependent. In terms of your second question, no, we haven't done that, but we can consider that.
Okay, thank you.
Thank you.
All right. Thank you. Thank you, Maria.
Thank you.
I would like to thank all the speakers for the fantastic session today, and also the audience for asking all the questions and made my life simpler. And I think we are taking a break for a coffee, and there are some posters too. Please wander around and speak to the ECRs and talk about their science. So we'll assemble back at 11:40. Thank you.
Hi there. So should we kick off with this next and final session? So my name's Chris Sibley. I'm a research fellow here at University of Edinburgh, and I'm an RNA biologist, so from a different angle from some of the talks which you're going to be hearing about today. I unfortunately wasn't here to make it for Richard's opening slides and the first 6 who were starting the Euan MacDonald Centre. But since I've joined, I've really seen something fantastic here in Edinburgh, something which is really unique. And it's this perfect constellation of clinicians, basic research scientists, the support staff who are all making this centre sort of thrive in today's kind of community. And you can see that we're having an international impact. We're making a difference in the clinic, and it's something which is really wonderful to be part of. So it's a great legacy to a great man in Euan and his family. So thank you for your support right from the beginning, and also for all the people who've contributed all the way through the growth of Euan MacDonald Centre to where it is today and seeing it going forward. And from a personal perspective, one of the things which I think has really stood out from the last couple of days, as well as the research, is also this development of early careers researchers going through, who then go on to become leaders. So I was just speaking to Siddharthan about the assembly of the gang from back in the day, all the people who've gone through the group. And we're fortunate enough to be supporting a Euan MacDonald Centre PhD student at the moment, but I think this is a wonderful centre which really fosters growth of new group leaders for the future. So without further ado, I'm delighted to introduce Professor Sharon Abrahams. You may have noticed that she was one of the founding 6 on Richard Chester's slides. So really looking forward to hearing about your work. Sharon qualified as a clinical neuropsychologist in 1998. She then moved to Edinburgh in 2004 and was awarded a personal chair in neuropsychology in 2015. She has a wonderful history and kind of long-standing interest in neurodegenerative disorders, particularly motor neuron disease and FTD. And many of you have come across her work in kind of developing the ECAS scoring system for assessing behavioral and cognitive changes in patients. And this has been translated into many different languages. It's been implemented into clinical trials, and it's been a real driver for assessing how new drugs and treatments are improving patients. Sharon is also the associate editor of the Journal of Amyotrophic Lateral Sclerosis and Frontotemporal Degeneration, and she was awarded the Thruda Newcomb Prize from British Neuropsychological Society in 2022. So we really look forward to hearing your talk, and the floor's yours.
Thanks very much, Chris. Can you hear me? Good. It just makes you feel old when you hear all that, particularly being there from the outset. Okay, I'm going to get on. I hope you're feeling cognitively able this morning. So we're going to just look at people have heard of this, she's doing cognitive assessment. So I want to explain really why we do this and how. And in terms of why, we're going to look at understanding the disease, and I'm going to also try and bring out the clinical aspects, which will be important for us, and how, yes, I will, as usual, talk about the ECAS, if people are not too sick of it by now. So Caroline has mentioned already this idea of a spectrum, and if I can get, have I got my, oh yes, yeah. So I want to emphasize the fact that half of our people, I call it ALS or MND, all of the people that we've assessed over the years have ALS, the most common form of MND. I haven't done research into other forms of MND. So I want to emphasize this, in that half of the people have no problems with cognition and behavior. It does seem to be restricted to a motor neuron disease. And we're quite, it's well established now that 5 to 15% have a type of dementia, and this type is called frontotemporal dementia. It's very different from Alzheimer's disease, as you'll see. With Alzheimer's, the first thing you see is many memory problems, and we hardly see that. Here we see problems mostly with behavior and some other functions. Now, there are 3 types of FTD that happen in the general population without MND, and the one we see with MND is more specific, and it is the behavioral variant. So what we were particularly interested in over the years is the spectrum, and this mid-range of the spectrum. The rest of people, there's about a third of people who are showing some cognitive problems or behavioral problems, and Caroline has already mentioned this diagnosis of ALS with cognitive impairment or behavioral impairment, and I'll talk a bit more about how we know that. So the first thing I want to highlight is the types of behavior change. People sometimes don't quite grasp this. One of the primary symptoms is disinhibition, and this is quite hard on carers and relatives, and it can be socially inappropriate behavior. So it can be something minor. It could be, I had a person who was taking his shoes and socks off in a restaurant. It's quite minor, wow. But it's not appropriate, and they were embarrassed by it. And it can be more extreme. It can be to sexual disinhibition, inappropriate comments to people. The second really prevalent symptom is apathy, and I'm going to tell you more about that later, how we've investigated it. But the best way to learn things is to talk to people who have lived experience of the disease, and I'll just read this out. This is from a person with ALS's wife, and I didn't know them. She'd heard my talk and wrote this, and I asked for permission to use it for teaching. So I'll read it out. My own GP has told me that ALS just affects the limbs and not the brain functions. So that's wrong on many levels. I have to disagree, as in 1 year of my late husband's life, I saw the change of a caring, conscientious family man into an uncaring, unsympathetic, confused and aggressive person that none of his family and friends recognized. A professional man that did the most bizarre things that were totally out of character. So we have a personality change. And even colleagues had gone, he'd noticed that he'd gone from a very efficient team leader to a couldn't care less attitude even before he was diagnosed. And this very much relates to Caroline's data on these pre-symptomatic changes. It's possible that those cognitive and behavior changes are happening well before the motor changes. He became unrecognizable, like a stranger, even though we'd been married for 22 years. It's touching. You can imagine the feeling. And I always say, next week, I've been married for 25. And my husband isn't the same person that I married. I'm not the same woman that he married. And it's quite hard to dissociate that what is abnormal behavior change from what is, you know, developing relationship, how you are over age. But one of the most telling things that she said was the professionals he saw did not, in their appointments, see the man he'd become, because we get a snapshot of someone. They walk in and say, oh, they're very friendly, and oh, they're very funny. And then you speak to the person's relative who's with you, and they say, well, they weren't like that. They were very reserved. They're now being flirtatious. And you don't see it until you ask someone, speak to people, to see the change. Now, cognitively, what's going on? And as has already been mentioned, we have a change in what we call executive functions. So these are the higher order functions, planning, etc. And I always use this analogy. Imagine Sunday morning, after more than 2 whiskies, you wake up and you think, oh my gosh, I've invited the in-laws over for Sunday lunch. And the mist clears and you say, it's fine, I've planned. I've got all the shopping in, I've got the menu. And you wake up, go downstairs, and you say, oh right, I've got a lovely roast. You've left it in the freezer. It's too big to defrost. You can't get it in the microwave. You're not going to put it in the bath. I've heard people do that. But so what are you going to do? And you think, well, it's okay. I've got some lovely sea bass in the freezer as well. I can defrost that in time. So you problem solve until you find a solution. But then it's also about timekeeping, organizing yourself. You have to do so many things at once in order for it to all, 2 o'clock, I've got a beautiful table laid out and then can impress my in-laws. So there's lots of multitasking, cooking, potatoes, chopping salads, dealing with the kids who are trying to kill each other in the next room. So it's a lot going on. And I use this example because certainly with FTD developing, it's one of the things people often report. Well, there were some changes in the cooking. It was the potatoes weren't done. And they were so usually so proud of their dinners that they produced. Okay, so how do we assess these things? Now, as has been mentioned already, there's this test called letter fluency. And I know over the years I've harped on about this, but it is a very sensitive measure, not only for ALS, we get it, I think the most prominent deficit in people with PSP, and so in Parkinson's disease. And so I'll explain how we assess it. Now, it's about saying as many words as you can, beginning with the letter. So as you can see, ship, shore, snake. But the problem with ALS, some people have problems speaking or writing. So we have to take that into account. And what we did, this is a few years ago, we developed something called the Verbal Fluency Index, in that after they'd finished the test, later on in the interview, I got them to write out or say the words again, repeat them. And that gave me an estimate of how long it took the motor component. And you take that out of the actual equation, and you're left with an estimate of how long are they thinking of each word. So here's some data, actual data, cognitive, how exciting. If I fall off the stage, I'm so sorry. But, okay, so this is how long they've taken to think of each word. So this is an example. This one was a 5-minute test of S words, and you can see a healthy person producing loads of words. This one was written. And this is our person with ALS. There's lots of words there, but they have slowed writing. So once we've taken out how fast they were writing, and we've got an average of how long it took them to think of each word, we're left with here 10 seconds up to 60. Now the average is here, as you can see, and it's about the same. Most people are doing like a healthy control, but our problem is here. These people who are taking a long time, and here, these people, it's 60 seconds to generate one word. Now, that's a long time. And when you're assessing someone clinically, you think, well, maybe they've forgotten their instructions, and you say it again, and they'll go, no, no, I know what I'm meant to be doing. It's that generation, that thinking of a new word. So over the years, we've when we first presented, they said, oh, it's nothing, it's not pathology, they're tired, or, you know. So we showed it with brain imaging, and this is Laura Goldstein, who's a psychologist at King's, and Nigel Lee, who really pushed this field forward. As a neurologist, he was one of the few people that used to stand up and go, verbal fluency, look at this. And so it was great. He was very embracing, and I did PET scanning. And to be honest, I didn't even know what motor neuron disease was when I took this job. I was attracted by the fact that they said, oh, we'll teach you how to scan. And it was very new then, early 90s. And so with PET, what you can do is you can stand next to someone, you can present information, then we don't get such movement artifacts as MRI. So it actually had lots of positives about it. And we asked people to generate words whilst they were in the scanner. And what you have here, if I can get this working again, so this is reduced blood flow in certain areas of the brain, and you can see here dorsolateral prefrontal cortex reduced during fluency bilaterally. And to keep up with the times, we also did fMRI later, and again showing pretty widespread, but particularly frontal, reduced activation when people are producing. And so we said, well, look, it's evidence of dysfunction and probable pathology. I'm going to show one more cognitive. We're going to get this one at this, and then I finish with the heavy cognition. So again, people say, well, they're just slow at producing. It's just they produce this processing things slowly, which you do find in some other disorders, such as Parkinson's disease. And so I got together with a really great cognitive psychologist, Bob Logie, and who developed this task of dual tasking, how we do two things at once. So we combined this test at the top, which is a processing speed test. And you can see on the first image, what flashes up on the screen is an image with two arms. And you just have to press the button or say which arm is longer. So it's very easy. But when you flash it up at 17 milliseconds, it's not that easy. And healthy people start making errors. We can combine that with another task, so it splits people's attention. And the task we combined it with was just remembering some numbers. So you present some numbers, you say, I'm going to ask you at the end of the test, you do the visual inspection time task, and then you ask for the numbers. So they are actually paying attention to two things at once. And what we found that our ALS patients show dual task impairment, and what happens is when they do two things at once, there was a lot more errors on each individual task than people without ALS. But actually, they were processing the information as fast as anyone else. So that, which arm is longer, they were absolutely like healthy controls. So it seemed to be very executive, this splitting attention. And clinically, it's useful in terms of when people are doing something, do one thing at a time, don't present too much. Simple stuff. Now I'm going to move on to apathy, which is an odd concept. It's about not being motivated. We're all apathetic sometimes, not being motivated to do something. The picture is of Ratko Radakovich, who came to me as an MSc student. And as you can see, that's him at Potato Land in Dublin. So the idea is you make sure you get the most unapathetic person to do this research. And we developed together a scale. And we tried to take out the motor, the physical disability from the scale. And we looked at 3 types of apathy. Executive apathy. So that is, I've got an example, yes, I'm not able to focus on a task until it's finished. I get easily confused when doing several things at once. Initiation apathy, which was, I can think of new things. I try new things. I set goals for myself. And the last one is emotional apathy. And this is just not being engaged emotionally. I'm concerned about how others feel, so it's the opposite. And before I do something, I think about how others would feel about it. And you know a lot of people who are that have that kind of emotional response to people. And what Ratko showed, it was a specific type of apathy. And as you can see here, it's the initiation apathy. It's about generating ideas. What should I do? And the other two types of apathy was not so much. And as we predicted, it correlated with verbal fluency. Both of them are these problems with generating ideas. And as you can see here, verbal fluency score here, higher score, better verbal fluency. Higher score on apathy is the more the apathy. And it's pretty good correlation there. Okay, what do we do with all this? I'm going to move on really to how. So along came Thomas Bach. I think he's somewhere here in the audience. Yes, hello, Thomas. And I can see you up here. You look very noble in this picture. It may have been a while ago. And, and the... You've had a hard night there, I can see. And Judy Newton, who I'm not sure she's here today, who is a wonderful clinical nurse, MND specialist, and who leads the whole nurses in Scotland now, consultant lead, something like that. And she at the time came up to me, so this is all very interesting. I want to do a master's with you. And so there were challenges, and I thought, this is perfect. All these neurologists, all these years have told me, give me a test I can use in the clinic. And I avoided it because we're psychologists, it's 2 hours to get a good profile. And so, okay, Judy came along, nurse specialist, we'll develop something. And here is how the ECAS was born. It was mixed blessings developing the ECAS because we developed it, launched it, and it went crazy because people didn't have a test. So particularly about amongst the Europeans, 28 languages now, very quickly translated it and using it in their own countries. So I'm going to give you a little flavor. We've got a lovely website. You can look more if you're interested. I think Caroline mentioned something about domains, multi-domain. I wanted to keep it broad and not just assess the functions that I know they're going to have problems with. I kept it broad to assess memory and visuospatial, which are more affected in Alzheimer's disease, because people come along, they're an older group of people in general, and they may have MCI to do with Alzheimer's, so I wanted to distinguish it. We designed it for physical disability, so you can do it written, spoken, and you can switch in between. It's fine. And there is this behavior interview. Speak to a person who knows them well and get some impression of how things may have changed. Very briefly, you can see how cute it is. There's a lot of development in this with Judy. We've got a naming task. We've got some comprehension, which is, for example, which of these fly, I think it is, or which can you fly in? I can't see. There's some spelling tasks, which as we've been picking up from our speech and language therapists, there's spelling issues coming along. There's good old letter fluency. I put 2 in. I knew it was sensitive, so it's free fluency, S words, but also a fixed fluency, which is say as many 4-letter words beginning with T as you can. So it's much more, let's search the lexicon, and really much heavier on the retrieval. Now, unlike other screens, we put a lot of executive functions in. Backward digits, there's a trails task, which you have to go 1A, 2B, 3C on. There's an inhibitory sentence completion. I'll just show you a bit more of this because it's incredibly sensitive to FTD. So this is a task where you give people a sentence, the last word is missing, and you ask them, Not to complete it with a sensible word, I want the word you give me to be completely unconnected to the sentence in every way. It's not as easy as it sounds, because automatically you think of the sensible completion. You have to inhibit that, generate something else. Now, this is a patient who didn't do so well, so it's the postman knocked on the, you're going to think door, letterbox you think of, and you want to say something like orange, something completely unconnected. As I say, they didn't do so well. It's a sensible completion. They'll get no marks for it. He brought his umbrella with him in case of. Sensible is rain. The person said snow, far too related, so they will get a 1 mark for that. So it's not as easy as people think. Now, as I say, there are some memory and visuospatial tests, and we've shown that these are sensitive to Alzheimer's disease, and in fact, we can distinguish between Alzheimer's and the cognitive impairment in ALS by looking at these two more frontal functions and more posterior brain functions. And just to show it works, this is in our Scottish patients, and the areas that we have an impairment are executive, fluency, and language, as we predict. Less so for memory, less so for visuospatial. But one of the things we're interested in, well, how does this relate to the motor symptoms? So we looked at disease stage, and this is the King's disease staging system, and not to go into too much detail, but it generally, the stages will progress in terms of the region of involvement. So you have 1 region, 2 regions, and stage 4 is when people need respiratory help or gastrostomy. And what we found was, so these are Z scores on the left, and so the lower the score, the worse the performance. And what we found, yes, it seemed to relate to disease stage, and particularly stages 3 and 4, people didn't do so well cognitively. And there was an increase in behaviour abnormalities as people got to the later stages of the disease. Now, one of my favourites, I still kept on standing up to these places, and they said, but there's no pathology in the brain related to these other areas. I had that many times. And so one of my favourite studies, it took us years to get this. Colin Smith, I know he was here yesterday, set up the brain donation tissue bank for ALS. And this is Jenna Gregory, who did the wonderful analysis of this tissue, who's up in Aberdeen. And we wanted to know, well, let's look in these other areas to see if there's any pathology. I did have people saying to me, well, I've looked in the hippocampus, there's nothing there, this is not real. I said, look, mate, you're looking in the wrong area. And so we went for these very specific areas, if I can show you. So I, with Colin, said, well, look, you've got to look in areas, these are the dorsolateral prefrontal cortex, and we divided those areas up in terms of Brodmann's areas based on the neuropsychology that we've known. And so we, as I say, language, fluency, we chose the areas. And Jenna was looking at TDP43 abnormalities, so that build-up of those proteins. And we found that people who had done the ECAS when they were alive, and it related very well to the protein abnormalities. So all people who had done not so well on the ECAS had these protein abnormalities, and it matched to the areas that we predicted. So the 3 with executive dysfunction had the abnormalities in the dorsolateral prefrontal cortex, etc. And every single of our patients with an ECAS problem in life had these abnormalities in areas of the prefrontal cortex. So it was my kind of, look, we're right, it is something. So I'm going to end this really by talking about something very clinical and how these changes do impact people's daily life by looking at I've had 3 studies looking at decision making, and in particular in related to mental capacity. Do people have mental capacity to consent to things? We looked at clinical trials and treatment with gastrostomy. Now, mental capacity is divided into a number of processes, and you have to show that each process you can do in order to pass a capacity assessment. You have to understand the information that people give you. You have to remember it. You have to be able to weigh up by thinking of the pros and cons of each choice and then weigh them up. And finally, you have to communicate the decision. So you have to be able to communicate. And we generally assume legally people have capacity unless we demonstrate otherwise. And capacity isn't one general thing, it's time and decision specific. So you have to show it for each decision. And we were showing problems with consent to clinical trials. And in our study looking at consent to feeding tubes, gastrostomy, Helena found that quite a few people had problems in the understanding, in particular, of the information we were giving them. So let me just say of blue, right? So 9 of those people had problems with understanding, some in reasoning, some in both. And in fact, 43% of our group would have failed a capacity assessment. I was quite shocked by that because I kind of did it saying, oh, I'm going to show they've all got capacity and this is not a problem for you. But Helena, we developed some support, some decisional supports. So I said, look, it's the information we're giving them. Generally, it's this massive text. And so we said, okay, let's develop an information sheet easier to understand. Put pictures in, divided it up into bullet points, as you can see here. And then not only that, I did something classical. Make a decision table, comparison table. We do it at home, right? What are the pros? What are the cons? So that it helps you balance them. And with that, we went from our 43% with impaired to just 4. Almost all people could pass an assessment. So I was delighted about that. And as I say, it helps people retain autonomy in their decision. So I'm moving on to a controversial subject, and we've got a new study just starting. You'll see Mario, who is here somewhere. I can see him over there. I've just employed him. He's going to run the whole thing for us. And it's funded by the MNDA. Thank you, Brian. And I know it was a brave step to take. People have avoided this subject for years, and for clinicians, it's going to hit them hard because I think there's no more neglecting it. As people would know, there is a bill, it's gone through already second reading, of assisted dying. And we know that there's been a lot of public attention from people with MND over the years. People have gone to the Court of Human Rights to try and fight and have always failed. Now, we're not taking a position on this. Oh, here we go. So we've got a group of us together, myself, Amma, from King's. Michael Cholby is a professor here of philosophy, and his speciality is into the ethics of dying. What a combination, that's it. And Zach Simmons is in Penn State in America, who's a specialist in quality of life. And so together we've put this together, and we're doing the study. And it will be about decision making. We are not making a stance. I'm reassuring Brian here. We're not making a stance like we're for. It's a very polarizing decision that to the public, oh, I'm against it, I'm for it. We're not making a stance. What I'm ensuring in the future is people are able to decide for themselves. So it can go either way. So someone who's just diagnosed may think, I don't want to go down that route. I'm going to do an advanced directive that I want assisted dying later on. So we would interview a person, do a capacity assessment, and just make sure that people had seen the pros and cons of each choice and been able to weigh them up. So they might decide, actually, I'm not doing that. We'll see how things go. So it can go either way, hence no stance. You got that, Brian, because you have to present that to the board. Okay, that's me. I know I've probably gone over. You can see all these lovely smiling faces are all a few of the people that have come through to be trained and have done all this work. And in particular, I want to thank all the people with MND and their families who have volunteered over the years. And in relation to Euan, I'm sure Euan, when I first presented this data, he had this look on his face of, what the hell is she talking about? And he then came round and said, no, I'm going to participate. She's got something, I'll participate. And from then, he participated in almost every study that we were running. It came to the stage where I told my researchers, don't recruit Euan, he's done enough, and he's skewing our data. He's just not, he was not an average person. And so, but he was wonderful in terms of accepting this kind of work and encouraging it, so I do thank him. I will leave it there, as I'm sure I've run over, and we've got wonderful early careers now, haven't we? Okay, thank you very much.
Thank you. That was a great talk. Do we have any questions from the audience? Go on in a minute.
Thank you for the great talk. My question relates to the capacity experiment. So essentially you said it's important that memory and weighing out are given in a person when they're sort of trying to go through these tasks. But then you say that only the DLPFC is affected. No. So I'm wondering if there's areas such as the ventral prefrontal cortex are involved in these things.
Yeah, so it's not only the DLPFC, it's just I seem to pick that one up quite a lot in terms of scanning and in terms of cognitively. Now, you can get memory problems by having a frontal lobe damage. We know the right frontal lobe is very much involved in retrieval of memory. And also, your attention is poor, so you're not taking things in as much. Unlike Alzheimer's, whatever you take in, you should be retaining it over time. With Alzheimer's, you actually lose the information. With other disorders, frontal lobe, you keep that information, but you're not taking in enough. So it's not only, and it's a slightly different types of memory problems. People are always like, well, where's your hippocampal damage then? But memory is not unitary, as I'm sure you know. Does that answer you? Kind of.
It's more complex than just one specific area related. Thank you.
Any more questions? In the middle.
Thanks so much for your interesting and accessible talk. One of your slides, you were talking about regions that were affected by motor neuron disease and how this, the more you had, the more likely it was you were going to have ECAS, problems showing up in ECAS. Does it matter which regions are affected by motor neuron disease, or is it just how many?
Well, we had this association, so people with language dysfunction had language affected, executive had executive errors, but we have to remember the brain doesn't really work like that. It isn't one area which is solely responsible for one function. It's network. However, it is a case of you're disrupting the network with protein abnormalities or cell death, and you're going to produce that symptoms. So it matters, but it's also, someone was talking to me and they said, is it like nodes that are connected? And I suppose that's an acceptable model. So you've got different areas, but the connections are the important element. And I think we showed DTI data that actually showed the white matter fibers being degraded in those areas, ones going to the frontal lobes.
One more question down here.
Thank you. Lovely talk. Loved it. So this may be a very naive question, but how do you deal with cultural differences and non-native speakers, especially when it comes to certain behavioral aspects and, of course, the language per se? And you don't have a baseline, right, to tell whether that's deteriorating or not.
It's a great question and a problem for all neuropsychology. Yeah, we try and interview people to say, well, what change has there been? This idea of context is very different culturally. In some cultures, a woman shouldn't stand next to a man. And I've had that with people, and they say, well, that's, when you speak to them, no, that's not done in my culture. So you have to bear that in mind. Even between families, what's acceptable in one is not acceptable in another. So I know someone said to me, oh, my father came down with only his underwear on, and I thought, I'm thinking, he had his underwear on, what's the problem? But you know, it's very different, and you have to bear that in mind. The translations were a nightmare to do because each one had to be culturally adapted. We had to adapt the pictures, the sentences, and we do it by the local psychologist does it with us, so they have some experience. But those people who live in this country, who are, their primary language is a different language, it's a problem because we don't have that Anglo local culture. We can't have 20,000 ECASs to do. So it is an issue, and it's, I think, the subtlety of the interviewer to pick that up.
Thank you. If I can extend thanks to Sharon. And we're now kicking off with some more early career flash talks. And the first is Dr. Laura Comley, who is a postdoctoral researcher in the lab of Lindsay Murray. And she's going to be talking to us a little bit today about how axonal innovation can shape spinal muscular atrophy.
Thank you. Afternoon, everyone, and thank you so much to the organisers for the opportunity to tell you a bit about my work looking at intramuscular axons today. So I'm going to change tack a little bit and talk about spinal muscular atrophy, and the nice thing about going near the end is that a lot of these topics have already been covered earlier in the symposium. But spinal muscular atrophy is a motor neuron disease which primarily affects children and young people. And it's caused by mutations in the really aptly named survival motor neuron gene 1. And this leads to low levels of SMN protein in the body. And there are now 3 treatment options available for SMA. And these have wildly changed the outlook for children with SMA and their families. However, there are still a lot of motor problems even following treatment. I've pulled this stat from a clinical trial looking at a gene therapy for SMA, but I really could have taken it from any one of them. The story is the same across all of them. And this is really a gold standard. These children were diagnosed by newborn screening. Treatment was initiated by, on average, 20 days of age. So it's very, very early, pre-symptomatic, though perhaps we should be calling it clinically silent. And even with this really early, really efficient therapy, you can see 36% of children still had problems with gross motor skills. And these are going to be things like being able to sit up, being able to swallow. They're not minor things. And we're really interested in why these deficits are still remaining in so many of these patients. So we are using mouse models of SMA to study this and to look at the neuromuscular system in this mouse model following treatment. And our hope is the more that we can understand the pathobiology of SMA, then the better we can design next generation therapies so that we can improve the outcome for more children. So in order to understand what treatment is affecting and where the deficits are still remaining, we first obviously have to have a good understanding of what the pathology looks like prior to treatment. So we use a mouse model called the Delta 7 mouse model of SMA, and these mice live to only about 12 days of age. And specifically, we use this group of muscles called the cranial muscles. These have got a lot of benefits for my work. They're very thin and very flat, and we can hole mount the whole muscle on a slide and look at the entire innervation pattern. And that means we can extract a whole lot of information about the incoming axons, about what the end plates look like on the motor fibers, and we can get a really detailed look at it. The other good thing about these muscles is, as Tom Gillingwater touched on yesterday, selective vulnerability is a thing in this mouse model. So, as he was saying, even muscles side by side in the body are not going to react in the same way. These 4 cranial muscles all react in different ways to the disease, so that gives us a really good tool. So I'm going to initially show you some data from this green muscle, the LALC, and we know this one is selectively very vulnerable. So we quantified innervation at the neuromuscular junction in this muscle across time. So we looked from day 1 until day 12, and we looked at the incidence of vacant end plates and partially occupied end plates indicating neuromuscular pathology. And what we found was on day 5, we had a significant loss of innervation. This is entirely what we expected. We've looked at this muscle so many times. It's early. Neuromuscular pathology is early. It's coming in at day 5. So we then, because we can see the whole innervation pattern, we can count the number of axons in the incoming intramuscular axon bundle. So I then counted axons from day 1 to day 12. And we were really surprised to see actually in this muscle, on day 1 of life, we've already lost about 40% of axons innervating this muscle. And it's continuous, and by day 12, the situation is even worse. So axonal loss is actually happening very early, prior to loss of innervation at the neuromuscular junction. So the obvious next question is, well, then when we treat, what happens? Can we improve the situation? So we gave these mice an analog of one of the approved therapies, and we initiated treatment at either day 2 or day 3. And basically, there was no improvement in axon number. The axons are lost at day 1. If you treat at day 2 or day 3, you're not improving the situation. And that could be the end of the story there, right? Axons are lost early. By the time we treat, it's too late. But the selective vulnerability really comes to the fore here. So we obviously looked at all of our muscles, and I don't have time to show you the data from all of them. But if we take another very severely vulnerable muscle, this is the AS muscle, this one actually didn't have a significant loss of axons until day 3. So then the story changes. When we treat at day 2, that is early enough to give a significant improvement in axon number compared to an untreated SMA mouse. If we delay by 24 hours, it's too late, they've gone. And then we don't have any improvement in axon number. So early treatment can preserve axon numbers, but it has to be initiated prior to the loss of these axons. So hopefully I've convinced you in this whistle-stop tour that intramuscular axons are vulnerable in SMA and are lost very early, and the early treatment can preserve them, but it does have to be prior to the loss of them. And every muscle is on its own trajectory there, so it's really important to consider which muscles are being used in studies designed to look at improvements with therapy. And the next steps then are to try and develop combination therapies, so we can give an SMN upregulator, but we can also give something that's going to help support axons and hopefully eventually make things better for more patients. So thank you so much. There's the Murray, oh no, sorry, I didn't give you that slide. But there's the Murray Lab, and thank you to the Euan MacDonald Centre for their support and all our funders, and happy to take questions.
A super quick question. What do we know about the reasons why different muscles have different trajectories?
Both a lot and still not the answer. We really don't know why selective vulnerability exists. Many, many different things have been looked at, like fiber type, like fast synapsing, delayed synapsing, where they are in the muscle. There's nothing that we have found so far in decades of research that can really pin down why some are vulnerable and some are resistant. It's $1 million question.
So you show the axonal loss. Is there associated, the neurons themselves, are they lost as well, or are they still there to be treated and promote regeneration?
Do you mean like centrally in the cell bodies?
Yes.
At this time point in this mouse, I don't think anybody has specifically looked, but Charlotte Sumner, who's an incredible clinician in the States, has done a study on patient postmortem tissue and mice where they looked at the ventral roots very early on, and they also saw loss of motor axons much, much more proximally, and they saw changes in the maturation rate as well. So it suggests that the problem is all the way along the axons. Just Thank you so much.
So our next speaker is Dr. Shreya Sharma, who is a postdoctoral researcher here in Edinburgh in the labs of and she's interested in the mechanisms underlying glutamate-excited toxicity using stem cell models.
Thank you for organizing this, and thank you for letting me, giving me the opportunity to discuss my work with you. So as Chris mentioned, I'm going to be talking about glutamate-mediated excitotoxicity in ALS. So glutamate-mediated excitotoxicity is a type of neuronal injury where due to excess glutamate, the postsynaptic neuron is overactivated, and that leads to its death. And this pathway is especially interesting because riluzole, which is so far the only globally approved drug for ALS, targets this pathway. But as we all know, it's quite ineffective. Hence, it is necessary to look at this pathway in a greater detail to unravel more effective therapeutic targets. So glutamate-mediated excitotoxicity is primarily mediated by the AMPA receptors, which are a type of glutamate receptor. And AMPA receptors are of two types: calcium permeable AMPA receptors, through which calcium can pass through, and calcium impermeable AMPA receptors, which does not allow the passage of calcium through them. So what happens in ALS, and several studies, including studies from our lab, have shown that the specific vulnerability of lower motor neurons to ALS is due to their high expression of calcium permeable AMPA receptors, which leads to excess calcium influx into the motor neurons, which then sort of activates a cascade of events, eventually resulting in excitotoxicity. So one of the ways to look at the AMPA receptor and the current that passes through it is by whole cell patch clamp electrophysiology, which we have, it's not moving, sorry, is by whole cell patch clamp electrophysiology, where we patch onto a cell and then we can record the currents that pass through the patch of the membrane that we have in the recording pipette. And then what we do is we wash in AMPA, which is an agonist for the AMPA receptors. After washing in AMPA, we can see this, give it away. So we can see, after washing in AMPA, we can see a robust downward current, which is blocked when we have a calcium permeable specific blocker called as NASPAM. Now, for the purposes of this talk, the thing to remember is that greater the percentage of NASPAM block, that corresponds to more number of calcium permeable AMPA receptors on the surface. As a majority of patients with ALS have sporadic forms of ALS, one of the first questions that we asked was, is the AMPA-mediated excitotoxicity in any way relevant in sporadic ALS? To assess this, we took iPSC-derived motor neurons we took iPSC-derived lines and generated motor neurons from sporadic ALS patients, and then assessed the presence or absence of AMPA-mediated excitotoxicity. What we found was, of the 4 sporadic ALS lines, 2 of them show an increase in the calcium-permeable AMPA receptors. These 2 lines also show an increase in excitotoxicity because after challenge with AMPA, we see higher percentage of cell death. So from this data, we can say that a subset of sporadic ALS lines do show AMPA-mediated excitotoxicity. The next question here is this AMPA-mediated excitotoxicity in any way dependent on TDP-43, because both sporadic and patients both with sporadic and genetic forms of ALS show TDP-43 pathology, where TDP-43, which is a nuclear protein, exits out of the nucleus and starts to deposit in the cytoplasm. To look at that, we used motor neurons where TDP-43 has been knocked down, and we find the same phenotype, that there's an increase in the percentage of calcium permeable AMPA receptors, and there's an increase in excitotoxicity. What was very interesting was that in these neurons, when TDP-43 is re-expressed, we can rescue both of these phenotypes, indicating that this pathway is indeed mediated by TDP-43. Next, to further understand the molecular pathways that could be involved in this process, we undertook bulk proteomic and transcriptomics and found in the TDP-43 knockdown motor neurons, and found downregulation of this gene called as DPP6. This was interesting because DPP6 is a GWAS hit and has been implicated in ALS susceptibility. What does it do? It acts as an auxiliary subunit to the A-type potassium channel, and it regulates the activity of this potassium channel, thereby also regulating neuronal excitability. We've also seen that loss of TDP-43 leads to cryptic splicing of DPP6. I'm not going to go into a lot of details about this, but I'm willing to chat if anyone wants to know the details. What was even more interesting is in the 4 sporadic lines that I showed, two of the sporadic lines show DPP6 downregulation, and if you remember, these are the 2 sporadic lines which also have high calcium permeable AMPA receptor activity and excitotoxicity. So the next question was, if DPP6 is downregulated, what happens when we overexpress DPP6? And we did exactly that. After overexpressing DPP6 in the motor neurons, in the TDP-43 knockdown motor neurons, we then assessed the high calcium permeable AMPA phenotype and excitotoxicity, and we see that its overexpression can again rescue both of these phenotypes in the TDP-43 knockdown motor neurons, as well as in the sporadic ALS motor neurons, indicating that the AMPA-mediated excitotoxicity is modulated by DPP6. To summarize, what I've shown you so far is that we do find AMPA-mediated excitotoxicity in sporadic ALS, and this pathway is dependent on TDP-43. What I've also shown you is that a gene called as DPP6 is downregulated in the TDP-43 knockdown motor neurons and a subset of sporadic ALS. And overexpression of DPP6 reverses the high calcium permeable AMPA phenotype and the AMPA-mediated excitotoxicity. What we're interested to do in the future is to look at the extent of heterogeneity in the DPP6 expression in sporadic ALS, and also see if we can find SNPs in the DPP6 locus. This is in collaboration with Professor Valentina from Cardiff and Dominic, who's a clinical PhD in the lab, and in the Anne Rowling Clinic. The other thing that we want to do is develop splice switching antisense oligonucleotides against the DPP6 cryptic exon and see if we can use it as a therapeutic modality. I would really like to thank my lab and also the stem cell facility, which is headed by Karen. And I would like to thank David Story and all the people involved in this project. And thank you so much for listening. And if you want more
We have time for one quick question if anyone would like to ask. I was going to ask, do you see the same change in this particular gene in cryptic splicing in familial ALS lines?
We've not looked at the familial ALS lines, but we do have lines with TDP-43 mutations, and our plan is to look at the DPP6 expression there.
That's great. Thank you very much. So our final flash talk is Dr. Daniel Jutzi, who trained as an RNA biologist in Bern in Switzerland. He then moved over to London with Mark David Rupp, and where he kind of developed this fascination with RNA binding proteins. And he's recently moved up here to Edinburgh University, where he's joined my lab, and he's going to be talking to you a little bit about his work on FUS and how it shapes the RNA binding landscape.
Thank you very much, Chris. Hi, everyone. Yeah, my name is Daniel. I'm a molecular RNA biologist, and I would like to tell you briefly about my recent work with the RNA binding protein FUS. So as you know, mutations in FUS are a rare but often aggressive cause of ALS that is predominant in young adults and teenagers, where FUS mutations account for up to 50% of cases. And what these mutations typically do is they induce mislocalization of FUS, which is normally a nuclear protein involved in RNA processing and the DNA damage response, to the cytoplasm, where it then aggregates and exerts an unknown toxic gain of function that leads to motor neuron loss. Besides two structured RNA binding domains that directly contact RNA, FUS also contains long intrinsically disordered regions that enable it to undergo condensation through liquid-liquid phase separation, a process that is similar to what happens in a lava lamp. However, how these two properties contribute to FUS function and its toxicity in ALS remains unclear, and understanding this will be important to inform on suitable therapeutic strategies. And so to dissect condensation and RNA binding experimentally, we set out to identify point mutations that selectively impair either process. And for the RNA binding mutations, we could really build on our previous structural work together with the ALA lab in Zurich and mutate 7 key residues that engage in direct and sequence-specific interactions with RNA. To inhibit condensation, we then tested 3 recombinant GFP-tagged FUS variants that we designed based on published studies. And as you can see, one of these variants with 7 tyrosines replaced by alanines completely failed to form fluorescent droplets in the test tube. And so we selected this as our condensation deficient mutant. But of course, we also wanted to know how these mutants behave in a cellular environment. And so to address this, we generated first some U2OS cell lines expressing either wild type or mutant GFP FUS from the endogenous locus, and therefore at physiological levels. And in these cells, what we found is that wild-type FUS partitioned into these bright nuclear condensates that we later identified as paraspeckles, whereas our condensation-deficient mutant clearly is not enriched in these structures. And likewise, after laser-induced DNA damage, we observe a fast recruitment of wild-type FUS to DNA-damaged foci. And again, this recruitment is clearly impaired by the condensation mutations, but not by the RNA binding mutations. And so together, these findings really validate this mutant, and so we turned our attention next to what happens to RNA binding. And so by mapping FUS RNA crosslinks in a transcriptome-wide scale, what we found is that all FUS variants still bound to pre-mRNA in a very widespread manner, and mostly to introns. However, zooming in, we could also really identify specific sites or also larger regions where binding was clearly dependent on either the structured RNA binding domains or also the condensation capacity of FUS. And we think that at least some of these changes could be explained by alterations in sequence specificity of the mutants. And so in line with the literature, we found that wild-type FUS preferably bound to G-rich motifs, but surprisingly, we also detected clear enrichment of many C-rich motifs, which had not been described before. And while, as expected, the RNA binding mutations strongly impaired specificity towards both type of motifs, our condensation-deficient mutant selectively lost specificity towards the G-rich motifs, whereas binding to C-rich motifs was relatively unaffected. And so, taken together, we conclude that the structured RNA binding domains of FUS confer specificity towards both G-rich and C-rich motifs, whereas condensation somehow selectively enhances binding to G-rich motifs. And so after this extensive characterization and many more things I did not have time to show you today. What we are doing now is we are generating 4 mouse embryonic stem cell lines in animal models where we introduce these mutations alongside an aggressive ALS-linked mutation into the endogenous mouse FUS gene. And ultimately, this will allow us to determine whether loss of RNA binding or loss of condensation is able to rescue motor neuron loss in these animals. And in doing so, we hope to get some new insights into the disease mechanism of FUS-linked ALS. With this, I would like to acknowledge all the funding bodies that have contributed to this work, as well as all the people. And I want to conclude by thanking the organizers and the MacDonald family for this opportunity to present our work here. And it has been, for me, a very inspiring and especially yesterday afternoon, also moving conference. And thank you very much.
Thank you, Daniel. Do we have any questions?
Hi there. With the RNA binding mutation that still forms paraspeckles, are the paraspeckles structurally intact, or is there something?
We didn't look at ultrastructure of these paraspeckles, so we didn't do any super resolution imaging. It could be. So, I mean, these structures are built on a long non-coding RNA that FUS is also known to bind. And so mutating the RNA binding domains would presumably affect these interactions.
Yeah, because they have that lovely donut structure that you can see in situ.
Yeah.
Okay, thank you.
One more question on the far side. Yeah.
The FUS condensation leading to movement to the paraspeckles and regions of DNA repair or DNA damage, is that implying then that there is increase in DNA damage and any recruitment of DNA repair?
Sorry, can you say that again?
Well, so that you're getting recruitment to these paraspeckles. So are there any changes in DNA repair mechanisms in those?
Yes, absolutely. Yeah.
And how are you determining that?
Yeah, so we've actually found that having these point mutants has been really, they've proven to be a really good tools to dissect how FUS acts in the DNA damage response. We've looked at recruitment of downstream factors. HDAC1 is one of them, SFPQ is one of them. So if FUS cannot condense appropriately, then the recruitment of these downstream factors is also impaired. And at the same time, we could also identify that some defects that have been described upon FUS depletion or knockout before are also not linked to this condensation, but are rather linked to the RNA binding capacity of FUS that is required to maintain the expression of mRNAs encoding other proteins involved in the DNA damage response. So, yeah, in summary, I think our mutants have been really helpful in dissecting direct and indirect contributions.
Have you looked at phosphorylated H2X, H2AX?
Yeah, yeah, and 53BP1 as well. So we see in the condensation deficient cells, under basal conditions, there's a small increase in gamma H2AX. In the RNA binding deficient cells, when they are challenged, we see a very dramatic increase in 53BP1 foci. And that is one of the cases where we think it's an indirect consequence of gene expression changes.
Lovely, thank you.
That's great. Thank you very much, Daniel.
Thank you.
Finally, in the last talk of this session and the 2 days, it's my real pleasure to welcome Professor Max Palmer to the stage. Professor Palmer is the Professor of Medical Statistics and Epidemiology at UCL. He's also the Director of both the MRC Clinical Trials Unit and the Institute of Clinical Trials and Methodology at UCL. And since joining the MRC in 1987, I believe, he's published in over 450 peer-reviewed articles. His clinical practice has certainly improved patient outcomes and changed the way clinical trials have been designed. And his methodological, I can't even say it now. His contributions have certainly shaped research practice. He has been a leadership in the MRC clinical trials unit since 2010. He's been involved in significant trials for both cancers, infectious diseases, neurogenetic diseases, and his remarkable contributions to the field were rewarded with an OBE in 2019, whilst he was also awarded the very prestigious Royal Statistical Society's Bradford Hill Medal in 2024. So welcome to Edinburgh, and we look forward to your talk.
Thank you very much. That just shows I'm quite old, I think. As I was just talking to Siddharthan, and he was saying that this is like the last slot at Glastonbury, Elton John getting up and giving the last performance at Glastonbury. And I'd like to think that's the Legends slot, but I think it's more the seniors slot, isn't it? So I'd like to thank the organisers very much for allowing me to give this presentation, to be the last presenter at this wonderful meeting for the tribute to Euan MacDonald. It's quite an honour to give the last presentation for such an impressive individual. What I'll try and do is speak to you about improving health by improving trials, and talk to you about some of the new ways of thinking about trials. There's a little bit of repetition from what Savanka and Siddharthan were giving yesterday, but from a different perspective. And I think it's helpful actually to think about this in a more strategic way. What are we trying to achieve here, and why? I will also say to you very clearly that this will be a very non-technical talk, so hopefully most of you will be able to follow it, and just to get the principles and ideas across to you. So let's get started. Trials are all about asking a question. So if you go back to when you were learning about clinical trials, we've all learned about clinical trials in your training. You'll be, say, you ask one question, you answer the question in the trial. You have one primary outcome measure, you have one question, you answer the question in the trial. So what we're asking usually is, does this new intervention improve outcomes for this disease? So that's what a trial typically does, whether it's phase 2 or phase 3 trial. That's what it's aiming to do. The challenge is that process can take up to 20 years from the first initiation out of the lab of a new entity up to actual use in practice. And cost, if you talk to the companies, about the order of £2 billion. And despite all of that activity taking that long, for many diseases, and I've just given a few there, we've made little or no progress for decades, absolute decades and decades. And we've heard that about motor neuron disease, but motor neuron disease isn't the only disease. There are lots of other diseases we've made little or no progress for decades. I've given you some there, some other neurodegenerative diseases, some cancers, some broader diseases, some mental health diseases, advanced lung and kidney disease. And that list goes on. Many diseases can fall into this category. And so what's been the problem? The problem has been this, is that we've often had lots of phase 2 signals. So our phase 2 trials, which are relatively small, trying to get an activity signal for a new intervention. Say, are we hitting these biological targets? Are we making a difference in biology with this intervention? We get lots of those. What happens is then we put them into these large-scale phase 3 trials. Those signals that we've seen biologically do not translate into improved outcomes for patients. And we see this time and again. That's what we're seeing. And the problem is that most of the phase 3 trials that do not show superiority in this setting are where all the action is in terms of the costs. There's 75% of all the evaluation costs of a new agent. They take a long time. They're much harder to do. They cost, in the academic world, many millions of pounds. In the industry world, they cost hundreds of millions of pounds. It's not unknown for these two things to cost 200, 300 million pounds for one single phase 3 trial. Often many hundreds, many thousands of patients, many, many sites, many, many years. Now, I know what I'm talking about, because I've been doing phase 3 trials for nearly 40 years now. So I spent most of my life doing these things and knowing, actually, getting a sense how challenging they are, how long they take, and how disappointed we are at the end of these things. So actually, we've got the question wrong. We shouldn't be asking, does this specific intervention improve outcomes for this disease? We changed the question. The most important question is, how do we improve outcomes for this disease? That's what we're about. We're not really desperately interested in that specific intervention. So actually, once you start thinking in that way, just changing the question opens up so many different ways of thinking. So if you say, well, how do we improve outcomes as quickly as possible? Well, it's obvious, isn't it? You know that if a single intervention is likely to fail, what you do is evaluate many interventions or ask many questions all at one time. You would do that. That's obvious. You know where you're consistently failing, despite succeeding at the phase 2 stage, consistently failing at the phase 3, you include a phase 3 stage. And you have a seamless move from phase 2 to phase 3. You acknowledge that many interventions will not succeed. History tells us that. Those who do not learn from history keep replaying it, of course. So we know that many will not succeed. So let's just be aware of that and adapt. We know that science and knowledge will emerge as we go, both from within the study and out with the study. So we adapt the protocol as we go, from data within and outside of the trial. Now, if you were a standard scientist approaching this problem of how do we improve outcomes as quickly as possible, you would say those are the basic tenets you would attach to any scientific process, actually. But, of course, trials, historically, and mostly university, do not do that. They don't use any of those tenets. They have one question, do not adapt as they go, don't have a seamless phase 2, phase 3 component, and they don't acknowledge that most of those interventions will fail. So this has led to this idea of multi-arm, multi-stage platform trials, basket, factorial mouse. I'll give you 3 examples of how this way of thinking, which is barn door obvious, changes how you do things and what you might do. So let's deal with multi-arm, multi-stage platform trials. We've had Savanka and Siddharthan both present this very, very clearly, more clearly than I could have presented, the idea behind this. So I won't spend a long time on this. But just to say, this is just putting all of the research arms in one protocol, starting a number of them all together rather than sequentially, and then adding and dropping arms as you go, as data emerges. So that's a very simple idea. And going back to the principles which I showed you just here, very simple principles applied there. What is important for us to know as an academic community, as actually we as an academic community have a unique opportunity here to do something which actually nobody else can do. Industry at any given time is only interested in a single treatment for a given disease. For obvious reasons, they don't have competing treatments for the same disease at the same time. And a single commercial partner doesn't. It would be of no relevance to them to do that. Academia can look across the piece, though, and has an opportunity to look at many interventions all at the same time for that given disease. And actually, academia, as a consequence, is uniquely placed to do such trials. And experience supports this. Unfortunately, most of the applications have been just in phase 2. And people are presenting these things as new ideas in phase 2. That will not change the game, because we'll still have the problem that those phase 2 signals that we get out of that fancy new approach in the phase 2 setting of testing many things at one time against a single control will still get those signals not translating into improvement in patient outcomes at the phase 3 stage. So they will not solve the problem. And the reason people are presenting it in that sort of way is two. One could be because they can do it. They do what they can do rather than what they should do. And secondly, because it allows you to get results in the timescales, which allows us all to fill our CVs. It's not going to change the game in a major way. Let's give you an example how you can change the game. Savanka has mentioned this in his presentation yesterday. This is Stampede, where we first started this thinking in about 2000. Now it's about 25 years ago we started the thinking, and it's been running for 20 years. It's probably the first modern phase 3 platform trial, and at 20 years now, the longest running platform trial. So just to give you a sense, I won't take a long time on this, just to give you a sense of what the situation was at that time. In 2000, we'd had men with prostate cancer and only had hormone therapy as their treatment for 40 years. Nothing had changed for 40 years, 4 decades. Median survival was for men with metastatic disease about 3 years, and we launched Stampede in 2005. And to be a multi-arm, multi-stage phase 3 platform trials, having many arms, adding and dropping arms. And through that time, in the 20 years, we've evaluated chemotherapy, bisphosphonates, a COX-2 inhibitor, radiotherapy, the new ARSIs, such as abiraterone, metformin, estrogens. What we've done is look at old drugs, repurposed drugs, new drugs, lots of other repurposed agents. I mean, estrogens come from hormone replacement therapy used for women to drive up the estrogens, reduce the testosterone levels. Radiotherapy has been evaluated in this trial as well. We've collaborated with 5 different companies, and this is the trial. And I won't spend a great deal of time on it. It's the standard of care right at the top, and the various arms. We started with 5 research arms, B through to F, and added arms G, H, J, K, L at various times over that period of time. It's been a huge collaboration. More than 12,000 participants, hundreds of researchers around the country in both the UK and Switzerland. 5 changes of standard of care during the process of over 20 years, 4 directly from Stampede itself. So we've changed the standard of care directly as a consequence of the results of Stampede on 4 occasions. And the most important thing is survival now has moved from 3 years median for men with prostate cancer, metastatic prostate cancer, to 8 years. It's a big change. If you think of any other way you're going to do that, you would not do that in 20 years. You would take 2 generations to get there, probably. So the prize here is huge. Once you throw out the standard way of doing trials, you can think, actually, what other adaptations can we do? We've dropped research arms, we've added research arms, we've changed the control in research arms, typically by adding successful interventions from the trial itself to be the new standard of care. We've changed the primary outcome measure because science has changed. We've changed the sample size. We've split the population into subgroups because there are targeted therapies coming through. So all of that has happened, and you can make those changes while retaining the scientific integrity of the trial. A complete change in thinking of how you do trials and how you prosecute trials. Actually, if you look at MND SMART, it's already done this. It's dropped research arms, it's adding research arms, it's in the way of changing its primary outcome measure. Of course, the big prize is the third one. You change the control and research arms by adding successful interventions, change the standard of care. More than 100 randomized trials using this sort of platform design has been used, or some variation of it. And of course, it was very, very successful during the COVID pandemic. We've taken this thinking further, and I'd like to give you 2 more examples of actually, once you start opening your mind about asking many questions as possible at once and being adaptive as you go, you can actually do incredible things. And I'll give you an example of one project that we're just starting now. with colleagues in Birmingham. So this is a problem of wound infection after surgery. Wound infection after surgery is one of the most common infections worldwide. Huge, huge problem worldwide. It's a major issue for the patient, a major issue for health services. Even if we could reduce the infection rates after surgical procedures by a small amount, that would be a huge gain worldwide, because you think about the amount of surgery that goes on in the UK and then multiply that worldwide, it's huge. Little progress has been made in this area for decades. That's because there's a poor evidence base. You look at the WHO, Centre for Disease and Control in the States, NICE here, there's conflicting recommendations, there's only generic guidance. So we've developed this trial, so this is the basket factorial MAMS trial, and I'll explain it. So again, this ideas are very, very simple. Taking a number of different surgical settings. Here we've got neurosurgery, breast surgery, vascular groin surgery, cardiac surgery, obstetric surgery, and major lower limb amputations. All different types of surgery. All suffer from wound infections after it. You can see, if you can read it, the infection rates in each of those. What we're doing is evaluating a number of interventions, starting with 3 in each of those surgical settings. All in one protocol, this is. Each of those surgical settings. Some of those interventions overlap between surgical settings. So, for example, some of the breast cancer interventions or breast surgery interventions made the same as the obstetrics surgical interventions to prevent wound infection. And at various points in time, we'll see whether actually we'll do interim lack of benefit analysis. Is it worth carrying on with that intervention? And can we learn across the different baskets, across the different surgical settings about an intervention, both if it's failing in one area, should we remove it from other areas? If it's succeeding in one area, should we then transfer it to other areas? And you get over time, you get a modified set of interventions emerging. This is a phase 3 trial for each individual basket. One other thing I should say, of course, for some of those areas like neurosurgery and major lower limb amputation, we will never achieve the sample sizes that you would need for typical phase 3 trials. You would never do that. But we can still get some evidence in those areas and use evidence from the other surgical settings to help support the evidence of use of those interventions for those areas which are rarer in nature. So there are multiple trials across the different body types, one overarching protocol. This has been funded by the NIHR in the UK. It's a 5-year project with 26,000 participants. 6 companies are collaborating in it. So you can see, once you start opening up this way of thinking, you can actually take on really quite major issues and be quite ambitious in what you're trying to achieve. And changing the game in this area over 5 years will be a real major development. We're developing this sort of idea in a number of areas and now are working with groups right around the world in cancer, infectious diseases, mental health, psychosis. You see the 4 neurodegenerative diseases that were mentioned yesterday, progressive MS, MND, Parkinson's disease and dementia are in there, to develop this different way of thinking more strategically about what you might do and do things in timescales that we would never have dreamed of before. I'd like to just finish with how we're thinking even further for the future, just to give you an example of a different sort of design, which uses the same sorts of principles. So in some clinical situations, what we have is a number of treatments that are being used. There's not one standard of care, but some patients can take some of the treatments, partly because some of the treatments are contraindicated or they're not available locally, they're not available in that country or that region. So one example is how should we treat carbapenem-resistant infections in adults and children? A particularly important question in low and middle income countries, but also in other countries. There are at least 13 candidate regimens for this. Most are in use, depending on local practice. None is a universal standard of care. and many patients will be contraindicated for several of the treatments. And I'll give you an example of that in a moment. So this is what we've designed, is this the practical design for this setting. Each participant is randomized between those therapies or those interventions, those antibiotics, which they're eligible for. And the principle is getting randomized to the evidence wherever you can. So let me just give you some examples there. There's the 13 regimens on the left-hand side. Patient 1 has a history of a heart attack, so they're only eligible for 10 of them. They're eligible for 10 of them, ineligible for 3 of them. Patient 2 has got different criteria, as ineligible for how many of them are there? 6 of them. Similarly, patient 3 is ineligible for 4 of them. So you can see, actually, as each patient comes in, you can see a different randomization list for them. So what you aim to do for each patient with a specific set of characteristics, the aim of this trial design is to rank the treatments which are potentially possible for them. Can we rank them in some fashion? And there's an analogy for those who are more technically minded with the network meta-analysis. And we produce a single ranking at the end of this trial of all of the treatments and choose the best treatment for each patient for which they're eligible and available to them. So you can see this idea of testing many things as once and adapting as you go can be applied in this sort of setting as well. It's been applied in the carbapenem-resistant infections in adults. That trial is just ongoing, being run in neonatal infections. One interesting area is we've developed it in the snakebite area. Snakebite is a big problem worldwide because when patients turn up with a snakebite, often the doctor will not know what the snake has bitten them. So they'll have to choose from a range of different interventions. So working with Wellcome, we designed this trial, and that's just started using this practical design. There are other designs, but I thought I'd just give you 3 today, which give you a feel for some of the more innovative thinking in this area and how we're going to have to think and do differently if we're going to make progress in a number of areas in our lifetimes. These things are challenging to design, develop, fund, and run. You need some sort of 5 to 20-year time frame that you're looking at here, involving many thousands of participants in the study and many hundreds of researchers. But it's really, I hope I persuaded you that they're more than worth the prize at the end of it. What's been important is patient involvement throughout their process. In fact, some of them would not have got through the funding process if patients had not put their thoughts and support behind them. And it's heartening to see that at least some funders have recognized their importance. Increasing number of funders are doing it now. And a patient partner, I'll just finish by saying a patient partner given me this quote, is why don't you do all your trials in this way? And we haven't got a sensible answer to that question. And I'll stop there. Thank you very much.
Thanks very much, Max. I think you've confirmed that this was Glastonbury pyramid stage, main event, legend slot. Thank you. That was fantastic. Questions? There's Adrian.
Thank you very much for this interesting talk. You make a really compelling case for changing the structure of the way we do trials. My question was on, do you know of any trials that use this structure that compare pharmacological and non-pharmacological interventions such as bioelectronic medicines?
Yes, so in Stampede, we've had both radiotherapy and pharmaceutical medicines. In other trials, we're using a variety of different approaches, both pharmacological and non-pharmacological. There's no reason why you can't combine them. What you want to do, though, actually, is they're not really competing. What you should do is aim to evaluate therapies with different mechanism of actions, because most of our diseases will need combination therapy to really get to cure or home runs. And so if you evaluate interventions with different mechanisms of action, what you can do is combine them. And that's the way we've got from 3 to 8 years so quickly in prostate cancer. We've combined the successful interventions because we strategically planned to do that. So they're not, why would you have Tweedledee and Tweedledum in the same trial? Why would you do that?
Go ahead, please.
Thanks for the talk. What's your view on the importance of biomarkers in phase 2 studies with respect to getting the right investigational product into a phase 3 study?
So, of course, they're important. I'm not sure they get us the right intervention into the phase 3 studies, because if they did, then we'd be much more successful in our phase 3 studies. What they do is tell us what things are unlikely to be effective very well. So they're a gatekeeper in telling us, actually, these things are not worth evaluating. They don't necessarily tell you, just because you show a result on a biomarker that the intervention has an effect on, that it's going to be successful in improving patient outcomes. Does that make sense? So as long as we are clear about both the benefits and the potential drawbacks of these things, we should, of course, use them. I'm not saying they're bad in their own right, because we have to use short-term markers to give stop-go decisions, absolutely. But we should be aware of the shortcomings of those stop-go decisions.
Any other questions? Jade?
Thank you so much for your talk. I'm just wondering, from your perspective, how can government and policy change to better support the move towards more innovative trials going forward?
Yeah, it's a good question, isn't it? So unfortunately, what they need to be able to do is to invest larger amounts of money in an individual trial and for longer periods of time. And of course, that's a challenge. What you need to look at is what's the strategy here, rather than the tactic of doing an individual trial. And it's not easy to get them to think like that. One simple thing is the grant funding envelope timeline. It's typically 3 to 5 years. Well, sometimes you need to plan for 10, 15, 20 years. How do you do that? So we've got to think differently, but it doesn't make any sense for the government not to think strategically, but I guess that happens in a number of areas, doesn't it? Oh, gosh.
Donald.
Are you sure I'm not keeping you from lunch?
Thank you. Thank you very much for your lecture, which is very, very, very interesting and challenging. And as you know, trials are taking place for MND with the single arm, one drug at a time. But I think there's a feeling that maybe we need the future may be in combining 2 drugs or whatever. What thoughts have you got in that? That makes the choice probably more difficult.
Yeah, no, I agree with you. Combinations will be the way to go. Usually on the route to getting to a combination, usually you see some evidence of a benefit of a single agent on patient outcomes. Not always, but usually you do. That's our experience across a range of diseases. So it's reasonable to add them one at a time, but strategically plan to combine them. And that would be a reason to use different types of therapies with different mechanisms of action in your different research arms, so you have the opportunity to combine them, because we know that's where we're probably going. You're absolutely right. Whether you jump to that combination immediately, that would be possible, but it would be potentially very big risk, but a potentially big reward.
Thank you very much. I'm going to close the questions there. I will say a few words in a moment, but on behalf of all of us, Max, thank you very much. So that is the last of the formal talks. I just want to close the meeting with a few words. They'll be brief. First of all, I want to thank, and there's many people, it's a long cast, so I'm not going to name people individually, who helped put together the last 2 days, which I think was a resounding success. So thank you all. I need to thank the sponsors. I managed to get to the Alamar and Axol stands and thank them again, but also SLS, Azenta, and Miltenyi Biotech. Thank you very much for supporting the meeting. We're very grateful. There were 3 people who judged posters, Matt, Helena, and Rachel, and they were judging them in order to give prizes. And there are 3 people who won prizes. Now, I don't know if you're here, but if you are, can you come to the front? That's Inga, Tatiana, and Cindy. By deduction, you must be Tatiana. Now we need to take a picture, don't we? Yeah. Okay, yeah, all 3. And then I'm coming back. Stop here. Okay. Thank you very much. And now finally, before you escape, and as you escape, there's a grab and go lunch. So do grab a bag. I don't know what's in it, but do grab it. I just want to make 2 final announcements. The first is, as a result of yesterday, particularly the public symposium element of this, which I thought was very successful, and certainly the feedback. Late last night, very late last night, I received a note confirming an anonymous gift of £1 million to further the work. To further the work of all of us, which is obviously important and terrific. And then finally, I'd like all of us, on behalf of the centre, on behalf of the community, to thank Louise and Donald, who have come again today. They're just there. I'd like you all to turn, look at them, and give them a round of applause. Thank you very much. That would have been excruciating for Donald and Louise. That is absolutely not their style, but I think that's correct, it's right and proper. Thank you all for joining us, for your contributions, particularly the speakers. Of course, I'm biased. I think it was a terrific 2 days, and we keep going. Thank you very much.